virtual conference, Pharmacy College at Mustansiriyah University. Actually, this online conference offered us a great opportunity to meet with respective experts in their field. Let me remind you that this panel I will share it with Dr. Afrah. This session followed by a break for 15 minutes, then followed by our presentation from our researcher that contribute in this conference. Let me introduce the first speaker in this day, Zainab Al-Bayati. She finished her bachelor and master degree in Baghdad University, while her PhD in pharmaceutical sciences in University of Nebraska. She's working now in the Faculty of Pharmaceutical Sciences at the University of Kansas for Medical Sciences. Her research is focused on drug discovery, development, and evaluation of novel molecules for treatment of drug abuse like nicotine, methamphetamine, cancer, bone infection, preclinical studies of adverse and toxic effects of new drugs. She has also extensive experience in research involving targeted drug delivery to the brain, bone, and cancer tissue. And she has a long expertise contribution, like more than 50 scientific conferences and more than 100 abstract contributed paper. And her talk for this day is pharmacokinetic analysis of opioid, Gabapentine co-drugs. You are very welcome, Dr. Zainab. The floor is yours now.
Thank you so much for this introduction. Good evening, everyone. Where is my slide? Okay. Why it is saying, okay. It's saying stop sharing.
It's now clear, Dr. Zainab.
Huh?
You can start, Dr. Zainab, but you start from the first slide. This is the last slide, acknowledgement slide.
Yeah, I don't see my slides. It say Zoom as sharing your screen.
Yeah, sharing now. We can see your slides.
I don't see anything. Yeah, I see it on your side, but not here. Just a minute, somebody is there. I did sharing, but it is not sharing.
No problem. I think I can see your slides. So
yeah,
I can
but
see
I
them.
don't see it.
Let me open this. You're sharing, I see. Where's the PowerPoint file?
My file?
Yeah, your PowerPoint.
My file?
Yeah.
Oh, I thought they are gonna.
No, you're gonna run the PowerPoint.
Oh, okay, I just closed it. That's fine.
Yeah.
Okay. Give me a second.
There you go.
Yeah, and you can share. I think you can share your screen and share the PowerPoint presentation. So I think that you might have X'd out of the Zoom. Here it is. So then let's share this. Let's share. Do you want it on this screen or this screen?
Wherever.
Okay. So we can share that screen. And then now they should be able to see your presentation.
We're seeing it already.
Yeah,
Okay.
it's clear now. You can start. Yeah.
Okay, thank you, Dr.
Yes.
Okay, good evening again. In the next few minutes, I'm going to walk you through my study entitled Pharmacokinetics of Cortex. So just a few words about Cortex and why do we need to synthesize Cortex. So these Cortex in particular are synthesized to treat pain. And as you know, pain is an unpleasant sensory and emotional experience associated with actual or potential tissue damage such as cutting, burning, etc. So opioid, and as you know, opioid and in particular codeine are very effective in treating pain. They are painkillers. So in this study, one of the opioid, which is codeine, was combined or conjugated to Gabapentine to produce Cortex. So 4 Cortex were synthesized and then tested for the stability, analgesic effect, and finally the pharmacokinetic. You know, the first step is to design and synthesize, then to study the stability in the GI tract because the drug is intended to be given orally. And as you know, orally, when a drug is given orally, it's going to face different pH conditions: acidic pH in the stomach, basic pH in the intestine, until it gets absorbed into the circulation. So the idea is to give the drug orally, passes intact through the GI tract, and then get absorbed into the circulation. Once the drug hits the circulation or the blood, we wanted to hydrolyze or fall apart to release the parent drugs because the co-drug is made of 2 parent drugs. So that's the idea of synthesizing co-drugs. And in this case, codeine, which is an opioid drug, is combined or conjugated with gabapentin. Four co-drugs were synthesized, you know, because not all of the synthesized co-drugs are going to pass intact into the GI tract. Not all of them are going to be effective as analgesic. Not all of them are going to have good bioavailability. And not all of them are going to hydrolyze when they hit the circulation. So that's just a brief idea about the work. So as I already mentioned, the usefulness of the Cortex strategy is to, excuse me, is to overcome the physical chemical properties of drugs, okay, like insolubility, like the drug is getting hydrolyzed, the drug is not absorbed, etc. Some of these physical chemical properties. So these co-drugs are synthesized to overcome the various barriers to drug formulation and delivery, such as poor or extreme aqueous solubility, chemical instability, insufficient absorption after oral absorption. So such as rapid pre-systemic metabolism, so etc. But this is a brief idea about why we need to synthesize Cortex. So I come to the aims of the study. The aims of our study was 4. As I said, first of all, to design and synthesize Cortex, not only one. And second, to test the stability because the drugs are intended to be given orally. So we need to test the stability of these drugs. Third, we need to evaluate these drugs. Maybe they are going to lose their analgesic activity when they are combined together. Finally, whoever passes all these specifications is going to be tested for pharmacokinetic effect. Not all of them. So I'll start with the first aim. Opioid gabapentin Cortex synthesis. And in this case, codeine is conjugated or combined with gabapentin. And 4 Cortex were synthesized. We took advantage of gabapentin because it has 2 functional groups, an amino group and a carboxylic group. So the codeine can be conjugated to the carboxylic acid to form an ester, or to the amino acid to form carbamate. Also, two pro-gabapentine functional groups or drugs were combined either to the carboxylic acid or to the amino group to form either ester or carbamate codrug. So these are the 4 drugs. I abbreviated these as codeine gabapentine codrug number 1, or CG number 1, CG number 2, which is carbamate, this one ester, and the third one is an ester, the fourth is also an ester. So the second aim is in vitro stability study of the codex. As I mentioned, we synthesize these drugs, but we need to test for this stability for the stability of these codex, because these codex are intended to be given orally. And orally, as I mentioned, they are going to face different pH conditions. In the stomach, the pH is 1.3 to 2, acidic pH. At the beginning of the intestine, it is slightly acidic, between like 4 to 5. And then intestine, it is basic, which is between 7 to 9. And then if the codrug passes through all these conditions, which we do it in vitro in buffers, then we do it again using simulated gastric fluid by using like the enzymes in the stomach, which is pepsin, by using the simulated intestinal fluid, using the enzyme in the intestine, and then after that we test these in plasma, and if it is intended for the brain because it has analgesic effect, then we have to test it in brain homogeneity as well. So that's the idea for testing the codrugs or any other drug intended to be given orally, to test it in buffers or chemical conditions, and in enzymatic conditions, and plasma, and in the brain. So this is these graphs or chromatograms, HPLC chromatograms, were taken. These are snapshots. So this is the chromatogram for the codrug number 1, and this is for codrug number 2, and this is for the codrug number 3 and 4. And it shows that the first one is, the first and second are for the parent drug, which are gabapentine and codeine. The third one is the for the codrug. And the same thing goes for the second chromatogram. The first 2 represent the parent drugs, codeine and gabapentine, and the third one represent the codrug. The third one is for the codeine gabapentine number 3 codrug. Again, the first 2 chromatograms represent the parent drugs, codeine and gabapentine, and the fourth one is the codrug. The fourth chromatogram represent the parent drugs, codeine and gabapentine, and the codrug number 4. So this is the in vitro stability, and this is repeated in the simulated gastric fluid. It contains the enzyme trypsin, and also in the simulated intestinal fluid. Then after that, it is tested in the plasma. So as I said, the idea is the drug has to pass through all the GI tract conditions, acidic and basic. And then when we test it in the plasma, we need it to fall apart or to cleave to release the parent drugs. We want the drug to get absorbed intact through the intestine, but once the drug hits the plasma, we need the drug to release the parent drugs. That's the idea, the ideal idea, but not all drugs intended for oral administration pass these conditions. So this is the second M, in vitro stability test of codrugs. We come to the third M. evaluation of analgesic activity or pain management. We use very simple procedure or test. There are many procedures to test the analgesic activity, but in our study, we use this, what is called Teleflex test. It is a simple idea. The animal, in this case is the rat, is trained, and then the tail is exposed to an intense beam of light. We wait until the tail becomes hot, and so the animal flicks the tail. That's why it is called Teleflex test. And we measure the latency period from the start of exposing the tail to the light until the animal flicks the tail. This is called the latency period. We do this before giving the drug and after giving the drug. And we measure this, and usually in seconds, and there is a cutoff time. We don't want the tail to get burned. If the animal was not experiencing any pain, then 30 seconds we cut off the light, and we don't want to burn the tail. Okay, so for this test, the Teleflex test, we used 3 doses. First of all, I just want to introduce you to this. So the y-axis is the Teleflex latency in seconds. The x-axis is the time in hours. The first curve, which is the biggest one, represent the dose 25 milligram per kilogram. The second dose is half the first dose, which is 12.5 milligram per kilogram. The third dose, which is 6.25, did not show good analgesic effect, and the latency period was only 8 hours. So these 3 doses of the co-drug number 4 was compared to a physical mixture. The physical mixture contains codeine and gabapentine, the parent compounds, we call them. And the physical mixture, just by mixing codeine and gabapentine, the dose was the molar equivalent to the 25 milligram per kilogram of the co-drug. We have to compare like apples to apples and the same dose. So as you can see, the dose of the co-drug, the highest dose, which is 25 milligram per kilogram, showed the most or the greater analgesic effect, and for a prolonged time. So as you can see, it is like for about 30 hours. And then we have the second dose, which is half the 25 milligram per kilogram, 12.5 milligram per kilogram. It shows the less analgesic effect, but again, it is much higher than the physical mixture. This is the physical mixture, the open triangle. So in summary, the co-drug number 4 showed, which is the N-ethoxycarbonyl gabapentine codeine co-drug, showed greater effectiveness or greater analgesic effect as tested by the Teleflex test. And also for the analgesic effect was for a prolonged period of time compared to the physical mixture. So we can say the summary, we can say that the co-drug codeine gabapentine number 4 passed this analgesic effect test. It is good. So the fourth aim, we come to the pharmacokinetics. And you know the pharmacokinetics, which is what the body does to the drug. It includes the absorption, distribution, metabolism, and excretion, what we refer to it as ADME. And this one shows the route of administration. We are interested in the oral route of administration because, as you know, patient doesn't like to go to the hospital to get injection when the time of the dose comes. Patients prefer to get the dose orally, and that's what we intended to administer CodeReg number 4. So this shows the concentration with the time, and this is the typical curve for an oral dose. It starts by absorption gradually and then decreases because of excretion. So in this case, rats were administered CodeReg number 4 orally. We used 2 doses because from previous the Teleflex test, the dose 6.25 did not show good analgesic effect. So we use only 2 doses. So this one, the curve to the left, again represent the concentration, the molar concentration in the y-axis, and versus the time in the x-axis in hours. The curve to the left represent the dose 12.5 milligram per kilogram, and the one to the right represent the dose 25 milligram per kilogram. And as you can see, the CodeReg, which is in this blue, is less than the one in the purple. So as I said, the idea is this was then again. I need to mention that rats were treated with these doses orally, and then blood was withdrawn from the rat after 5 minutes, 15 minutes, 30 minutes, 1 hour, 2 hours, 6 hours, 9 hours, and 24 hours for the oral dose. And the oral dose has to be compared to an IV dose, which was in this case 1 milligram, because you know the bioavailability of a drug in the after IV dosing is 100%, and we need to compare the oral dosing with the IV dosing. Again, after administering the drug IV, we sample from the rat after 5 minutes, 15 minutes, 30 minutes, 1 hour, 2 hours, 6 hours, and 9 hours. We didn't do 24 hours because, you know, with the IV route, the drug is excreted much rapidly than when it is given orally. But with the oral dose, we did 24 hours also. I come to the curve. So the one to the left represent the oral dose of 12.5 milligram per kilogram of the CodeReg, and the one to the right represent the dose of 25 milligram per kilogram oral dose of the CodeReg number 4. So as you can see, the CodeReg hydrolyzed to release codeine and N-ethoxycarbonyl Gabapentine, which is kind of the carbamate moiety of the Gabapentine. Okay, now codeine also is hydrolyzed to release some morphine. That's why we determine morphine, codeine, N-ethoxycarbonyl Gabapentine, and the CodeReg. So the CodeReg hydrolyzed slowly to release codeine and N-ethoxycarbonyl Gabapentine. Codeine, in turn, is metabolized slower to release morphine, and that's why it is we included the morphine here. And so the bottom line is the CodeReg, when absorbed into the circulation, when the CodeReg hits the blood, it cleaved to release the two parents drug, codeine and the carbamate moiety of Gabapentine. The carbamate moiety and ethoxycarbonyl Gabapentine also hydrolyze even slower to release gabapentine. So the idea of the codreg is fulfilled. This table shows the result of using the two doses, 25, 12.5, and it shows the AUC, Tmax, Cmax, and the half-life. And as you can see, the Tmax was attained within 3 hours for the codreg, but it took longer time for the parent drugs to be formed or produced in the blood. But as you can see, the codreg cleaved to release codeine and ethoxycarbonyl gabapentine, or the carbamate moiety, and the N-ethoxycarbonyl gabapentine cleaved slowly and release gabapentine and morphine, and that's what we need. It is like slowly released drug. So I just want to conclude the four aims of the study. A series of codregs, 4 codregs were synthesized. One of them is carbamate, and 3 are esters. So codregs 1, 2, 3, and 4. So the idea of designing and synthesizing codregs is achieved, and then the stability of the codregs was measured, and the analgesic effect of these codregs were also determined, and finally the pharmacokinetics of the codreg number 4 was tested. We did not test, or we did not work on the gabapentine or the codeine gabapentine codreg number 1 and also codreg number 2, because codreg number 1 was hydrolyzed very fast in the GI tract. So it did not fill the idea of stability when passing through the GI tract. So codeine, gabapentine, codreg number 1 was not included in further tests like the analgesic effect or pharmacokinetic. Codreg number 2, codeine gabapentine number 2, did not hydrolyze at all when it passed through the GI tract or when it hits the plasma in vitro. So again, this one was excluded. Codeine gabapentine number 3, which is codeine and acetyl gabapentine, did not hydrolyze much in the GI tract, but it hydrolyzed when we did the in vitro analysis in the plasma, but I did not include it here because of the limited time. Only codeine gabapentine number 4 fulfilled the specification of a drug when given orally. And so the pharmacokinetic study was performed on the N-ethoxycarbonyl gabapentine codeine, or codeine codreg number 4, and thus it can be predicted that after the codreg is absorbed into the systemic circulation, it will be hydrolyzed in the plasma to afford codeine and gabapentine protected gabapentine. So this is the idea of synthesizing codreg, and we need to know the stability of the drug in order to guide the chemistry for future synthesis to increase the stability of the compound when it passes or when it tested in the GI tract buffers, GI tract or chemical buffers, and when it tested in the simulated gastrointestinal fluid, and when it is tested in the plasma in vitro. We need to guide the chemical synthesis to improve the synthesis of the drug, and also to optimize the pharmacokinetic parameters of the drug, because once it passes through the GI tract, then we need to test the pharmacokinetics. So we need to optimize the pharmacokinetic, because if a drug is absorbed through the GI tract, then it is in the circulation. And if it is a co-drug, then we need it to cleave to release parent drugs, in this case, the codeine and gabapentine. And finally, I would like to acknowledge Dr. Peter Krug's, he is the chairman of the department, and Ujjwal Chakrabarty, he finished his PhD in our department. Elizabeth, she did the Teleflex Test. John May, he did some of the LCMS, and Manjula Sankara, she did the technical effect for the LCMS analysis. And I will be happy to answer any question.
Thank you, Dr. Zainab, for your valuable lecture, actually. Thank you very much. It's really interesting. It's really interesting. It's really, I'm interested in the slide about the results showing the area on the curve or the bioavailability of the oral doses of the Cortex that you test them. Yeah, thank you very much for showing us this. Dr. Zainab, can you explain us what's the difference between these co-drugs and the parent drugs? Because I noticed like there is like, if we talk about the mass balance, there's like a big difference between the parent drug and the co-drugs. Am I right?
Yes, yes. That's why I said we used the molar equivalent. The dose was in molar, and the results was also in moles. We use the molar equivalent to 25. So this, what we used was like, actually was 22.5 mix of the gabapentine and codeine. In order to compare it to the dose we used for the Cortex, it is the molar. It is not like the milligram. It is the molar. Based
So
on
for
the
that reason, showing different area on the curve in this paragraph or in this figure?
It should be the same. Like when I say it is the molar equivalent of the mixture, of the physical mixture, it means gabapentine and codeine concentration is similar to the Cortex dose. Yeah.
Okay.
Does this answer your question?
Yeah, it's kind of, yeah. But let me ask you about this figure also. It's really interesting. I don't know if you, when you say that you test this drug in plasma, do you do test for protein binding? Do you know how, if this co-drug is highly bound to plasma protein? Because I've noticed some changes in the elimination phase. It shows like more than compartment model. It likes maybe 3 compartment model. I noticed, yeah, I noticed that distribution phase, especially when you increase the dose to 25 meg, there is an increase in the area on the curve, specifically in the phase of the distribution. So does this related maybe to the protein binding, or maybe to the slow hydrolysis of the drug, of the co-drugs that you prepare?
Or maybe 2, or maybe both.
Maybe both.
Because as you know, when the drug gets absorbed into the circulation, it's going to distribute based on the physical chemical property of the drug. Some of it is going to distribute, some of it is going to be excreted, some of them is going to go to the brain, where it is the target tissue, because it is intended to treat pain. So it is all this. Some of the drug is distributed, some of the drug is excreted, some of this maybe protein binding, which is good, because once it gets bind to the proteins in the plasma, like albumin and other proteins, then it works as a slowly released drug. It is gonna release from the protein.
Slowly cleared. I don't know if you use some like software to analyze your data, like for your pharmacokinetic data, for example, the clearance. Can I know which kind of software that you use to analyze your data?
Yes, we always use the WinNanLin software, which is we call it the Phoenix.
Phoenix.
software.
Okay.
So we
Okay.
plot the concentration versus time. We
Then,
put the numbers in the software, then we
Mhm.
get the results in the table. We get the AUC, Tmax,
is it
Cmax.
free online software or should be registered and
We
pay
bought
for
it.
this?
We bought
Yeah,
it.
you bought it. Yes. Yeah. Yeah.
Yeah.
Yeah. Yeah.
We bought it.
So, do you prefer physiological model or pharmacokinetic or compartmental model? Because you talk about the enzymes.
Do I prefer the complemental? I would say it is, we usually, with such pharmacokinetic, with the limited number of rats, which is 6 rats per
Yeah.
each time point, we use non-complemental design or model. Non-complemental.
No compartment.
But
Okay.
it looks, as you said, it looks
Yeah.
like it is a 3 compartment.
Yeah, yeah, it showed me, and there is like an issue when you increase the concentration. Maybe there is a slow distribution, maybe it's kind of, I don't know. It's really interesting. It's very interesting actually, this figure. Yeah, yeah.
It is not very water soluble. It is not very water soluble. The
So
Cortex.
maybe, yeah, but still this is
Yeah,
blood.
that's why we dissolve it in 15% polyethylene glycol to increase the solubility. So if it is not very water soluble, then you
Yeah.
expect more lipid soluble. Then you expect more distribution.
Okay, okay.
Yeah.
It's really interesting. I don't know if there's a question. Maybe.
Yeah, I think I'm seeing Dr.
Yes.
An'am. She is raising her hand.
Yes.
Okay.
Yes.
Yes.
Thank you very much, Dr. Zainab.
You're welcome.
Very nice work. I would like to ask you about the safety of this combination. Both of these drugs have possibility to induce respiratory depression, for example. And combining these two drugs together, it might increase this possibility, even the possibility of accidental intake of these drugs. So what do you say about it?
I really couldn't hear you very well. I mean, could you just raise your voice, please? I couldn't hear you very well.
I'm asking about the safety of this drug combination.
Oh, okay. We have not reached
Both
to this
drugs
point.
may induce respiratory depression.
Of course, we need to test for safety. We have not, even the toxicity, we have not reached. It is just a preliminary experiment.
Okay, good.
Okay.
Thank you.
Okay, thank you. Is there any question?
Yeah, Doctor, I see Dr. Munder.
Dr. Manda, yes, please, Dr. Manda. Go ahead.
Good evening, everybody. Really, I have a comment, not a question, for Dr. Zainab. I have nearly the same idea or research printed in 2012 in Swiss and pharmaceutics journals about design synthesis and hydrolytic behavior of mutual products of some non-steroidal anti-inflammatory drugs with Gabapentine using a glycol spacer in order to avoid or to reduce the local irritation of non-steroidal carboxylic group on the GIT. So I use a spacer, which is glycol spacer, make ester with non-steroidal from one side and ester with Gabapentine through the it is carboxyl group from another behavior, and also study the hydrolysis behavior in two different pH in the stomach, comparable to the stomach and the intestine, and hydrolysis by taking a plasma sample, also using HPLC. I may exceed my thoughts and make a combination with some idea from your research. I would like to thank you for this nice idea, nice results, Dr. Zainab and co-workers. Thank you.
Oh, thank you for having me to this meeting. Very much appreciated.
Thank you very much. Thank you, Dr. Manda. Thank you, Dr. Zainab, for your valuable
Thank you.
talk, valuable information, and hope to see you again in our conference, maybe in a workshop together about the pharmacokinetic. Thank you very much. Thank you very much.
You're welcome. Thank you for
Thank
having
you.
me.
Thank you. Thank you. And now we'll transfer to Dr. Frah to present the second speaker in our conference.
Thank you, Dr. Nassar, for your presenting. Actually, I'm warmly welcome all the attendants to our third virtual conference. And particularly, it's my honor to introduce our second speaker, Dr. Brandon Fred. Dr. Brandon Fred is Assistant Professor of Pharmaceutical Sciences. And he received his PhD in pharmaceutical sciences with an emphasis in drug discovery and development from the University of Arizona in 2014. And he also received a postdoc in medicinal chemistry and pharmaceutics. He has lots of publication regarding his field of study in order to be successful, transferred his academic-based discoveries to pharmaceuticals companies for clinical development. He is also interested in identifying single agents therapies, which coupled with controlling multiple dysregulated pathways in cancer. And he's also a member in different society, which including American Chemical Society and American Thyroid Associate and American Association of Colleges of Pharmacy. And I really warmly welcome Dr. Brandon to present his presentation entitled of New Paradigms and Strategies for FLT, which is the famous-like tyrosine kinase 3 precision in medicines and acute myeloid leukemia, which is known as a kinase-related disease. Thank you very much for your participating in our conference, and now the time is for you to give your presentation. Thank you.
Great. Yes, thank you so much for the introduction, and thank you to everybody that set this conference up. I really appreciate getting the opportunity to present some of my research. And so I'm a medicinal chemist by trade. So this project or this presentation is going to be really presenting this problem from a medicinal chemist perspective. And although, right, based on this title, it seems, right, that we're signaling out a specific type of cancer. You can apply the strategies that we used in this presentation to just about any type of a cancer that's being treated with precision medicine. And then beyond that, this can also be expanded to stuff like infectious disease, where there's a propensity for resistance. So the same problem happens in cancer, where the precision medicine that's used is frequently has issues with the tumor developing resistance. And so we'll kind of go over what we've done to possibly improve that issue. And so my talk is going to be kind of in two different phases. We first are going to discuss, just current precision medicine strategies in AML, and then discuss this clinical candidate that we identified and kind of how it compares to current treatments and how this was developed to overcome issues with the current treatments that are available. So when you're thinking about precision medicine, it really has its kind of early development in the early 1900s. And there was this gentleman named Paul Ehrlich, and he was interested really back then. A lot of work was done with dyes, right? The pharmaceutical industry, a lot of chemistry behind that came from the dye industry. And he noticed that when you had a specific dye that was placed onto a cell, it would localize to a specific area of that cell. And so he came up with this idea of a magic bullet where, you know, a drug can go to a specific area in the body or in a cell that's causing the disease and somehow manipulate it in order to relieve the disease. And so the idea of one drug, one target, I think oversimplifies the complexity of something like cancer. Because within a cancer, there are multiple different types of mutations from cell to cell, and they all have a lot of issues in terms of the pathways that are dysregulated. And so it doesn't really seem like this idea of blocking one specific pathway in a tumor that uses many survival pathways is going to have sustained effect in causing the tumor to go into remission. So there's also been work done using polypharmacy, right, where you take a drug, two drugs that are active on different types of cell survival pathways that a tumor exploits in order to continue to grow. And that is a good way to get multiple pathways blocked. But the problem with this, especially with this example, you have two kinase inhibitors, right? They have overlapping toxicity, and it cannot, sometimes not very well tolerated for the patient, because they're also taking other medication as well to treat this, their tumor. And so this comes back to the idea of using one drug, but instead of one target, right, the new area, the new exciting area for precision medicine is coming up with ways for a single drug to block multiple pathways. Or instead of multiple pathways, as we'll see in this presentation, multiple mutations of a oncogene. And so doing this, you're able to kind of predict how the tumor will evolve. And with that knowledge, because we've seen patients relapse time and time again, especially on medicine for AML, for precision medicine, if you know the pathways or the mutations that the drug selects for, you can come up with medication to effectively block those. And so you can kind of increase the remission time that a patient experiences when taking a targeted therapy. So when you're thinking of acute myeloid leukemia, you should also be thinking about FLT3, because this is a cancer in which FLT3 is a driver oncogene in about 30% of all cases. So this is kind of an exception. A lot of cancers may have a driver oncogene, but it's much, much lower in terms of the percentage of patients that have that. And so with AML, a large amount of patients have a FLT3 mutation that drives the tumor. And also in AML, almost all of them have overexpression of FLT3 to an extent. And so this is definitely an important oncogene that's driving the proliferation of AML. And so what ends up happening when this develops, the FLT3 protein, which is a receptor tyrosine kinase, undergoes a mutation. And this mutation causes the enzyme to be constitutively active. And so it's always active, always on, and it's fueling cell survival pathways, which then increases cell proliferation, right? And it causes progression of cancer. And so the trick is, I guess the problem with this is it's not always just one mutation. There is a mutation called FLT3 ITD, which is the region of the receptor that's on the inside of the cell before the kinase domain. And that can undergo duplication events, which then renders the kinase constitutively active. So there's that mutation that can occur. And then there's additional point mutations that occur in the active site of the kinase, which can also drive constitutive activity. So you really have to be considering all of these types of mutations when you're developing the next generation of precision medicine, because AML will evolve when the precision medicine is administered to the patient. And you want to be able to effectively block the additional mutations that present. So this is kind of a general setup of how a patient would be treated with AML. And so I think the important thing to consider is this is not an exception, right? This is kind of the rule for a lot of different types of malignancies. You typically don't give, right, just a single treatment. I mean, there's radiation, there's surgery, and then there's also chemotherapy. And that's really trying to chop away at the heterogeneity within the tumor. So with AML at diagnosis, right, typically a patient undergoes, chemotherapy. And so when that occurs, patient goes into remission. And right, so it essentially, looks like the patient is not necessarily cured, but the disease has been kind of put in check. And then over time, right, the remission, depending from patient to patient, can turn into then a relapse. And during this next relapse, new clones that have different mutations pop up. And so this is going to be a problem when you treat that with a precision medicine therapy, with a tyrosine kinase inhibitor. Because if that inhibitor is not active on all of these new types of mutations, it's going to select, right, for those mutations to be able to then make up the majority of the tumor in the second remission. And so when you're looking at this, right, the area in red is the AMLs that just have that single ITD mutation. And then all of these other ones are more complex mutations, right, that a lot of the current precision medicine therapies don't have activity on. So when you treat with medication that's not active on these different types of mutations, you actually select for those mutations to then grow. So this puts the selection pressure on the tumor, just like natural selection. You select for the clones that are best fit to survive. And those are the ones that are resistant to the precision medicine. So it ends up causing the disease to evolve. So that can be a major problem. And success of precision medicine really relies on the drug being able to block as many types of different mutations that are present in the tumor. And that's going to really induce a robust remission, right? So the patient doesn't relapse after, sometimes even just a year of being on treatment. So this is the current, kind of the most important FLT3 inhibitors that have been in clinical trials. 2 of them have been approved. Gliteratinib was approved in March 2018, and midostaurin was actually the first small molecule approved in 2017. And so when you're looking at these 2 that were approved, the problem with midostaurin, as we'll see, it's a natural product derivative. And I'm not saying that's a problem, but it's based on staurosporin. And we know staurosporin is just a general kinase inhibitor. And so with midostaurin, as we'll see, it is very promiscuous in the kinome. And so that can limit, right, the maximum tolerated dose, because if you're blocking a lot of different types of kinases, that can then cause adverse events and off-target toxicity. And then the other one that's approved, gliteratinib, right, it's active on some of the mutations, but as the FLT3 undergoes additional mutations, right, you start to see a reduction in affinity, right? So it's not able to very effectively block some of these additional mutations. And when that occurs, right, it's going to then induce resistance, right? So the profile, that the drug profile is not active on a specific mutation, that mutation will then be able to grow, and the AML will relapse. So our question was, looking at midostaurin and seeing that from this kinase dendrogram that it's hitting or it's active on a lot of different types of families within the kinome. But it is effective in terms of being used as a therapy for AML, especially FLT3-driven AML. And our question was, is it possible to generate an inhibitor that is active on a variety of these different FLT3 mutants without having this kind of promiscuous profile in the kinome? Because really, when you're generating a drug to be active on mutants, you can inadvertently reduce its selectivity. And then you'll be kind of in the same problem where the promiscuous activity of the drug is really going to lower the amount of time the patient can be on it and the level that the drug can reach in the body. And so what we wanted to see, right, if we can kind of improve this and generate a molecule that can block multiple manifestations of the oncogene, right, and that's really going to help increase the durability of the response for a patient with AML. So in order to get started on this project, what we use in the lab are kinase fragment libraries. And so these are small, typically heterocyclic compounds that have been designed to have a region that can bind to the hinge of the kinase. So this is where adenine binds, right? Because ATP binds at that region. So the adenine warhead binds at the hinge. So we have different types of heterocycles that mimic that. And then there's also around the pocket, right, very lipophilic regions that the drug can go into to kind of improve selectivity and also improve affinity. And so from doing this type of screening, what we found was this small fragment that was able to inhibit the FLT3 kinase at an IC50 of about 90 nanomolar. And so we're very excited with this result because this is a relatively small molecule, right? And there's lots of ways that we can modify that in order to kind of improve the potency and also improve the ability for this to block multiple manifestations of the FLT3 oncogene as it mutates. And so with this fragment in hand, we just kind of did very brief analoging at that R1 position off of this chlorine. And for the synthetic chemists in the crowd, right, you can very easily use palladium catalysis to work off of that, to kind of grow the scaffold off that R1 position. And so we noticed that when we changed that position just with these few analogs, that the drug or this, the new analogs kind of lost activity. But what we did notice is we screened this parent fragment against this FLT3 point mutation, which is clinically relevant. And we saw that it was just about as active on that mutation as it was on FLT3. So this was suggesting to us, right, that this type of scaffold may have pretty good activity on additional mutations. What we wanted to know is it possible for us to further improve this, especially for its ability to inhibit the FLT3 kinase? And so what we looked at is how this fragment was binding to the FLT3 receptor. Right, so these molecules bind at the kinase domain. And this here, right, is present in every single kinase. It's called the hinge. And that's where adenine from ATP makes a hydrogen bond with the active site. And so this here is binding at that same region, at least it's predicted based off of computational modeling. And with FLT3, right, this is kind of a unique site that's present on, it's accessible with certain kinases, is this allosteric region. And when looking at this, right, that chlorine is oriented into that region. And so we thought that if we further analoged off that chlorine, we'd be able to access this allosteric pocket, which then would be able to increase the selectivity and potency of the molecule against FLT3. And so we then went to develop synthetic methodology that allowed us to access that pocket. And so I think for the slides I sent out, I kind of revised them, cut a lot of information out. So it may not be exactly the same if you're following from the slides I sent. So the synthesis was disclosed in the slides, but this is just showing you the results. And this here is the part of the scaffold that gets into that allosteric region. And so you can see, right, this is the candidate that we identified from this. You can see that it's very, very active on FLT3. So the IC50 on this, right, is very, very low nanomolar. So this was kind of promising, right, that we were taking a step in the right direction. What's even more important when you're thinking about off-target toxicity is that this scaffold, or at least this example, does not have very strong inhibition on VEGFR2 compared to FLT3. And so this was a very good thing that we identified because VEGFR2 in the clinic, when patients are on kinase inhibitors that are very active in VEGFR2, it can lead to cardiotoxicity, right? And when this occurs, right, it'll lower or decrease the therapeutic window. So that can then influence how much of the drug you can give and for how long you can give it. And so without having much affinity on VEGFR2, right, you can dose this drug to block the FLT3 oncogene, and then some of the off-target liabilities, you won't be inhibiting those until much, much higher than the therapeutic concentrations of this. Okay, so in order to kind of get to this point, right, we first identified this tractable fragment, right, that we then were able to analog and push this fragment into the allosteric site on the within the FLT3 kinase domain. And one thing that we discovered is this linker region here. This is a methylene. A lot of drugs actually use urea at this region. And so we used the methylene because it makes it then flexible. And what we found is that that flexible linker kind of allows the drug to shift its conformation depending on what mutation is present. So it makes it a lot more robust in terms of its ability to inhibit multiple types of FLT3 mutations. And so we then took this candidate and we wanted to test it against other medication that was used in the clinic or that, that is approved for clinical use for AML. And so we really compared our drug to other drugs by looking at, in this example, we're using a model that incorporates BAF3 cells. So these are murine B cells, and you can transfect these cells with different types of oncogenes. And these oncogenes then make the cell dependent on the oncogene for growth. And so this is a very good tool that you can use to really test selectivity and activity of the drug on different types of oncogene mutations. And so from this, you can see the molecule, right, that we identified as you generate these different types of mutations in the FLT3 kinase domain. you can see that this very well inhibits these at, more, typically at 10 nanomolar or less. And what's even more important for us, right, you can look at the cytotoxicity of the drug against the normal BAF3 cells that have not been transfected. And you can see that the dose that is basically causing impairment of growth for normal cells is significantly higher than the efficacy dose in order to inhibit the FLT3 mutations. Right, and when you compare that to some of the molecules that have been investigated in the clinic, right, you start to see that the selectivity profile of the drug that we developed seems to be more promising in terms of having selective inhibition of FLT3 and additional FLT3 mutants. So another thing that we screen this against are patient-derived AML cell lines. And so MV411 and MOM14, right, these both have FLT3 mutation. And then, right, these additional cell lines have additional mutations within those regions of the kinase domain. And so you can see, right, when comparing the activity of KV1 to some of these other approved, this one is approved. This one was in clinical trials for a while and never got approval. You can see that if you're able to block, right, these additional mutations in the FLT3 or any type of oncogene, right, you are able to, right, induce a longer remission. And so remember with midostaurin, although it's no data for this, but I'm certain that it's active on that, midostaurin is promiscuous within the kinome. And so that issue, right, can then lead to off-target toxicity. And then with, right, the kinome selectivity, and we'll see of KV1, it is very, very clean in terms of other kinases that it inhibits. It has very minimal off-target toxicity. And so we screened this molecule against the kinome, and we focused mainly on the tyrosine kinase family because that's where this drug is most active. And from the screen, what we saw, right, is it's active on a lot of these FLT3 mutations, as indicated here. And you can see, right, this was screened at 10 nanomolar, and the lower the number, the higher the affinity, that it seems to be broadly active against a variety of FLT3 mutations that are clinically relevant. And so we believe that this type of an approach can actually help extend remission time in AML, right? Because this, according to the kinase selectivity, is much more selective, which has been shown to be much more tolerated as you increase, better tolerated as you increase selectivity within the kinome. And so this here, I'm going to go over some quick xenograph studies. So the drug was dosed orally in all of these examples. And keep in mind, we're doing this in MV411 xenographs. So this is not a very good model in terms of modeling the heterogeneity of AMLs that are observed in the clinic, right? This is a cell line. And so you typically can see pretty robust efficacy. We were really doing this for proof of concept to kind of show that KV1 has in vivo activity. And so when we administered this drug, we saw almost immediately within a couple of days that the tumor shrunk down. And over the course of treatment, the tumor is shrunk down to undetectable levels. Now it's not gone. That's always the caveat with precision medicine. It doesn't completely remove the tumor. Typically only surgery can do that. And so if you, and we'll see when you remove this treatment, we'll see in the next slide, the tumor will rebound. But we're also really curious how well this drug worked in terms of treating larger tumor volume. And so given this medication, even at sub-milligram per kilogram amounts, right, the drug is very effective at reducing the proliferation of this specific AML cell line. And so we then let the tumors rebound again. In this instance, right, we let them rebound. And we added the drug again once it reached high volume. And we noticed that it was able to, it's a little bit more messy as we go out here, but we noticed it was able to then shrink back down the tumor volumes. And so I think something interesting that you can notice is that the duration to shrink the tumors actually increased the second time around. And this might be showing, right, that the MV411 cell line is evolving and selecting for different types of mutations, right, that this drug may not have as high a potency on, but it's still able to reduce the growth of, right, of the AML. It just takes a little bit longer. Now, we didn't genotype any of the tumors within this part of the study, right, but it kind of suggests that it's possible that this MV411 xenograft has evolved or mutated. So it's a little bit more stubborn when you introduce the medication again. So another thing that you always need to consider when you're doing a drug development project is the mechanism of action, kind of really supporting that in an in vivo setting. So this kind of establishes the pharmacodynamics of how this medication is working. And so in order to do this, we had the MOM14 tumors, which are patient-derived AML tumors, and they were grown in non-skid mice, and then the drug was given at a dose of 1 mg per kg orally. And so when you're looking at this, these are blots from the actual tumor in the mouse. And so what we're looking at here, comparing to vehicle, so we're looking at activation of the FLT3 receptor, right? So it autophosphorylates itself, and the drug will block that autophosphorylation event. So as you can see, even at 1 hour and all the way up to 24 hours, you see that there is almost no activated FLT3, suggesting, right, that this medication is able to effectively block, right, FLT3 from signaling within this in vivo setting. We also wanted to look at different types of mutations, right, that were present and kind of see how robust this drug was in blocking these additional point mutations that will pop up as the AML evolves. And so same thing we did, looking at activation of the receptor. And you can see compared to vehicle, right, this plot is a little less convincing, but you can see when you're looking at it that it does appear, right, that it is reducing activity or signaling in that mutation, right? So it's blocking the kinase. This has a lot better blot, I think, and you can kind of see a dose response compared to the vehicle, right? As you increase the concentration of the drug, you can see a more robust inhibition of the oncogene, right? And this is one of those point mutations that can sometimes render the inhibitor compromised in terms of its ability to block this. So it kind of shows, right, that this medication is able to effectively block these different types of point mutations, as well as the original oncogene that's present in the AML when it's diagnosed. So it kind of shows that it's more robust and active on a lot of different types of point mutations. So by the time we finished up a lot of this data, the FLT3 landscape was getting extremely crowded. And so unfortunately, we were never able to get funding to put this into clinical trials, just because the field kind of shifted, and we never were able to secure investment to get this project translated into the clinic. But that doesn't mean it failed, because I think it shows kind of a valuable lesson and progression in the field of precision medicine having to be now optimized for mutations that pop up. And like I said, this occurs in tumors, and it also occurs in infectious disease. So the lessons that we learned from this, where it can be applied to a lot of different therapeutic areas. And so I'd like to again thank you guys for letting me give this talk. I'd like to acknowledge funding sources, collaborators, so I can take any questions that you guys have.
Thank you very much, Dr. Brandon, for this valuable information of this presentation. And we are really interested in such publication and a presentation regarding cancers and leukemia. Now we are open a way to give questions regarding your presentation. If there is any of the audience have any question, Dr. Brandon will answer the question.
And so I think Mohammed has a question about fragment-based drug discovery. And so it's a little bit different than traditional fragment-based drug discovery, because a lot of the fragments that we have, they're a little bit bigger. And so what we do is generate different types of kind of unique heterocycles that can serve as the adenine binding heterocycle to mimic ATP. And then depending on if it's bicyclic, tricyclic, it'll augment kind of how it interacts in the pocket. And it can then, from the very beginning, you can find something that's more selective for a specific kinase. And then with that, you can model it and then figure out kind of how it's interacting at the pocket and in the binding site. And then you can generate analogs of that to then increase potency and possibly selectivity. So we really use this to identify kind of novel warheads that interact at that kinase domain that are then tractable for us to be able to then develop it into a larger drug molecule that has better selectivity and potency.
Yeah, actually, I have a question regarding the approval of using FLT3 targeted therapeutics. Is it the main cause or the consequences from using this inhibitor because maybe afraid of this inhibitor can make or produce a subsequent mutation? Is that the main cause?
Yeah, so typically the mutation within a tumor, right, even from cell to cell, right, as it divides, it's unique, right? Not, the genetic material as a tumor grows, it doesn't do a great job of copying everything exactly. And so when that happens, you kind of have heterogeneous mixture of a lot of different tumors within AML, for instance, that all have different mutations that are basically present, right, as the tumor evolves. So the drug basically puts a selection pressure on the tumor that then selects for mutations that are resistant to treatment. So that's kind of how the tumor evolves to precision medicine. Those mutations are typically present already, and the drug just puts a selection pressure on it for those mutations to then grow.
Okay, thank you very much, Dr. Brandon. If there is any question from the audience?
I believe Dr. Anam and Dr. Bahar have questions. Dr. Afrah?
Yes, please.
Thank you so much, Dr. Brandon, for this astonishing, really work. But do you think this hypothesis is applicable practically, clinically? Because as we all know that for each gene, we possibly might have hundreds of mutations. So do we have to make gene sequencing for each patient and then try to apply this treatment with them, or what could we do?
So the I guess one caveat, the mutations have to produce a viable kinase. So you do have kind of a lot of different possible mutations that can pop up, but you kind of are restricted, or I guess the cancer is restricted by, generating mutations that still render the kinase active, especially for this type of pathway. But yeah, I mean, in theory, each cancer is from your own unique genetic background. And so it's all going to be different from patient to patient. And that can make it incredibly difficult to treat. And so one ways that you try to get around that is to use a companion diagnostic where you can take a patient's tumor and analyze it for a mutation that the targeted therapy can block. And so then you can enrich the patients for specific mutations that the drug will be active on. And that's frequently done in something, in oncogene-driven lung cancers, where the amount of patients that have a mutation or a specific mutation is going to be much lower, because lung cancer is extremely heterogeneous as well. And so you have to use a companion diagnostic to select for the patients that have a mutation that the drug can target. But really, as you introduce a new drug, it very likely will select for new mutations that you didn't realize existed. And so you're kind of always having to improve the medication to kind of be able to block these additional mutations that are present. So that is going to be, I think, an ongoing issue when you're looking at precision medicine in oncology.
Right, thank you. Okay.
Thank you very much, Dr. Bandin, for your valuable information regarding your presentation today. We really appreciate your participate for this nice presentation for not only the scientists or researchers, also for the postgraduate students in Iraq. Thank you very much.
Thank you.
Thank you. Now I will transfer to Dr. Massa regarding to introduce the third speaker. Thank you very much.
Thank you. The third speaker in the second day of our conference, Dr. Lina Ali. She finished her bachelor and MSc in Jordan, and her PhD was in Pharmacy School of Nottingham University. She has a lot of skills and expertise, but her research interest was mostly in bioanalysis and biomarker discovery, particularly in women health disease like preeclampsia and breast cancer, using advanced analytical strategies such as mass spectrometry and infrared macrospectroscopy to aid understanding of biomedical problems such as preeclampsia, cancer, neurodegenerative disorder, infectious disease, metabolomics, and proteomic studies, qualitative and quantitative analysis of proteins like LC-MS, ELISA, and Western blotting. And she got the award from in 2020 L'Oreal UNESCO for Women in Science 11th Young Talent Program. And her talk for today, actually she has a long list of skills and expertise, but for today, the title of her talk is the use of metabolomics approach to investigate the cytotoxic effect of phospholipid gold nanorods in MCF7 cells. You are very welcome, Dr. Lina. The floor is yours now.
Thank you very much, Dr. Massar. It's really nice to see you again after such a long period. We were colleagues together at the University of Nottingham. I'm so glad that I get the chance to see you. Hopefully next time we see each other face to face.
Inshallah.
I would like to thank you for giving me the opportunity to participate in your conference and present my work entitled, can you see the slides at the beginning?
Yes, everything is clear. Go ahead, my dear.
Thank you so much. So I'm so happy to share with you my talk entitled The Use of Metabolomics Approach to Investigate the Cytotoxic Effect of Phospholipid Gold Nanorods in MCF7 breast cancer cells. So through my presentation, I'm going to talk about what do we mean by omics sciences. I will focus on metabolomics as a branch of omics sciences. I will take you through the general workflow in metabolomics, the main approaches used in metabolomics, and I will end with one of the applications of metabolomics from my research lab, which was investigating the cytotoxicity of gold nanorods in breast cancer, MCF7 cancer cells. So at the beginning, metabolomics is a branch of omics studies. So what do we mean by omics? Omics is a science that deals with the universal and the comprehensive analysis of biomolecules. So if you do this comprehensive analysis at the level of the DNA, then we call this science genomics. If you do it at the level of RNA, then we call it transcriptomics. If you do this comprehensive analysis or profiling at the level of a protein, then we call it proteomics. And if you do this comprehensive analysis at the level of the small molecules, metabolites, then we refer to this as metabolomics. So actually, metabolomics is receiving increasing interest during the last decade, as it represents the end stream of this omics sciences. And it is the most likely branch of omics to reflect changes in the phenotype, to reflect the changes as in the phenotype. It is widely used in biomarker discovery for so many diseases. Now, to focus more, what do we mean by metabolomics? So metabolomics is best defined as the global identification and the quantification of a small set of molecules. Normally, we are talking about molecules with a molecular weight less than 1,500 Dalton, okay? And these molecules are present in certain systems, mainly most of the time, certain biological system. These small molecules can be carbohydrates, sugars, nucleic acids, amino acids, lipids, and all secondary metabolites present in any biological system, whether we are talking about urine, tissues, plasma, cells. All of these small molecules can be quantified and identified in metabolomics. Metabolomics has so many wide applications. It's one of the most important applications for metabolomics is to provide novel insights into the underlying pathophysiological mechanisms of diseases. It is also used to monitor the progress of the disease, and it's also used to identify unique biomarkers, to identify new and novel drug targets for drug discovery purposes. Now, the experiments of metabolomics relies on the use of advanced analytical technology together with bioinformatics. So you have to have advanced technologies with bioinformatics. Now, the experimental designs in metabolomics study definitely depends on the aim of the study, the biological sample that you are going to study, the available resources. But in general, most metabolomics study, they follow typical workflow that involves similar steps, which I'm going to talk about them briefly. So normally you start with your biological samples. So a typical application of metabolomics to have 2 conditions or 3 conditions, and you want to compare between these conditions. So for example, you want to compare diseased with healthy. You want to compare treated with untreated. Something like this. You want to compare between different conditions or different data sets. So you will have your biological samples, whatever was the biological sample, you can apply whatever type of biological samples. As we said, 2 conditions, for example, treated, untreated, diseased, healthy. Then depends on your biological samples, metabolites should be extracted, and metabolite extraction, we have different protocols for metabolite extraction. Most of the time we use liquid biphasic extraction. Definitely the type of the extraction, you want to use the best extraction method that provide the highest sensitivity, that provide the most coverage for most of your metabolites in your sample. And definitely it should be compatible with your analytical workflow that you are going to use afterwards. So after extracting the metabolites, now these extracted metabolites will be profiled or will be analyzed using sophisticated analytical instruments. Typically, we have a two workflow in metabolomics. You can either use mass spectrometry or NMR. If you use most of the time with mass spectrometry, this mass spec is coupled with a separation technique, either liquid chromatography or GC. If we want to make a quick comparison between the use of mass spectrometry and NMR in metabolomic study, mass spectrometry, the application of mass spec provides superior advantage over NMR in terms of sensitivity and metabolome coverage. Now, after analyzing the data or metabolite profiling using the analytical workflow, these collected data or raw data will be properly processed according to certain softwares, and then metabolites will be identified based on using specialized software, and identified metabolites will undergo statistical analysis. Most of the time we use two types of statistical analysis in metabolomics. We use univariate analysis and multivariate analysis. Now, metabolites that will be significantly altered between the two conditions that you are comparing, these metabolites will be considered as biomarkers, and they will be subjected to pathway analysis in order to identify the altered or the dysregulated pathways involved in your disease or in your question, whatever you are asking about. So the end aim of any metabolomics experiment is to discover a biomarker. And what do you mean by biomarker? A biomarker is a molecule or an indicator that can be used in order to indicate specific biological process or certain a response to certain treatment. So if we are talking about pharmaceutical drugs or therapeutics, then the biomarker will be a biomarker that can be used to monitor the effect of your drug or the effect of your treatment. As you can see here, we have different types of biomarkers. We have something we call it diagnostic biomarkers, which are used for diagnosis purposes to aid in diagnosis. We have something we call it predictive biomarkers, which are used to predict the disease before actually the patient has the disease. So if you have a predictive biomarker, then in this case, you can provide, you can put your patient under monitoring before the deterioration of your condition. So we have different types of biomarkers. Normally, we have two main approaches in metabolomics. So the first approach, we call it untargeted or non-targeted global metabolomics approach. It's a screening approach, which means that you want to see everything in your sample. You want to see the whole picture. You are not targeting specific molecules. You want to see all the molecules, all the metabolites in your sample. You are doing screening and profiling. And this approach is mainly used for the biomarker discovery. The second approach, which we call it targeted approach, as you can see, you don't want to see everything in your sample. You know your molecules, and you just want to quantify specific metabolites in your biological sample. And this is referred to as targeted approach. So for biomarker discovery reason, then you have to go for screening approach. And later on, if you want to validate your biomarker, then you go for the targeted approach. This is normally what we do in metabolomics experiment. Now, for application, as we said, we have so many applications for metabolomics. In my research, I used a metabolomics workflow to investigate the pathophysiological mechanisms in diabetes and preeclampsia. I also used metabolomics in monitoring the effect of certain therapeutics in what is known, it's a special field of metabolomics known as pharmacometabolomics. So I used metabolomics to investigate the toxicity of dexamethasone. I also used metabolomics to monitor the effect of metformin in healthy subjects. But for this presentation, I'm going to focus on the use of metabolomics to investigate the cytotoxic effect of phospholipid gold nanorods in breast MCF7 cancer cells. So, in the previous study, we synthesized two gold nanorods. One modified with big moiety, and I will refer to this gold nanorods as big gold nanorods. And the other one, we introduced a phospholipid moiety, which is phospholipid gold nanorods. We investigated the effect of these two gold nanorods on MCF7 breast cancer cells. And we noticed that phospholipid gold nanorods showed more significant and a higher cytotoxic effect on MCF7 breast cancer cells than the big. And as you can see, at all concentration, this phospholipid gold nanorods was associated with higher cytotoxicity on MCF7. At that time, we didn't know how this gold nanorods is affecting the MCF7, why it's showing a higher cytotoxicity if we want to compare it to big. What are the mechanisms behind this cytotoxic effect of phospholipid gold nanorods? So in order to have insights into the altered biochemical pathways and metabolic pathways that is affecting the MCF7 cancer cells, and to know why phospholipid is associated with higher cytotoxicity than PIG, we did a metabolomics experiment. So the aim was to identify the dysregulated biochemical pathways that are altered upon treating the MCF7 cells with phospholipid gold nanorods, and to show why phospholipid gold nanorods is affecting the MCF7 cells more than the PIG gold nanorods. So we started with we followed the typical workflow for metabolomics experiment. We had MCF7 cells treated with sub-inhibitory concentration. We always use sub-inhibitory concentrations. Okay, so we had 3 conditions: phospholipid gold nanorods treated MCF7, big gold nanorods treated MCF7s, and control or untreated breast cancer cells. We did metabolite extraction. We used a biphasic liquid extraction, and the extracted metabolites were dried and were subjected to LC-MS analysis. High resolution, we used high resolution mass spec. We used the Q-exactive coupled to ultra performance liquid chromatography. The scan range was from 70 to 1,400 with a resolution of 50,000. Now, after that, the data were properly processed for peak picking and noise filtering and peak alignment. After that, we identified the metabolites. We used the human metabolome database for metabolites identification, and later on we did statistical analysis. As I said earlier, we used 2 types of statistical analysis, sophisticated statistical analysis. We used multivariate analysis using a platform known as SIMCA, and we used also univariate analysis. Now, for multivariate analysis, in order to consider the metabolite as a significantly altered metabolites, you use a VIP value, which refers to variable importance projection. VIP should be more than 1 in order to consider the metabolite significantly altered between the 3 groups. While for univariate analysis, we use what we call it corrected p-value, or false discovery rate less than 0.05. Now, metabolites that were found to be significantly altered in multivariate and univariate analysis were considered as biomarker, and these biomarkers were subjected to pathway analysis in order to identify the pathways that were dysregulated upon treating the cells with phospholipid or big gold nanorods. Now, for the results, as you can see, these are we applied 10 replicates from each condition. Now, for LC-MS analysis, we were able to detect 6,936 mass ions. Out of these detected mass ions, 747 metabolites were putatively matched or were putatively identified. The identified metabolites were involved in different pathways. They were involved in amino acids metabolism, lipid biosynthesis, and the nucleotide metabolism. Now, for multivariate analysis, we used OBLSDA plot, or orthogonal partial least square discriminative analysis. As you can see in the OBLSDA plot, you can see obvious and evident separation and clustering of the 3 conditions. So this obvious and evident separation and clustering of the 3 groups indicates that there is a huge number of metabolites that were significantly disturbed or perturbed between the 3 conditions. when we want to extract these metabolites that were responsible for this obvious separation using the BIP value more than 1, and we found that 661 mass ions were altered. Out of these 661 altered mass ions, 179 metabolites were identified and were significantly altered in the multivariate analysis. Now, for univariate analysis, we used, as I said, we used ANOVA, since we are comparing 3 groups, and we used a corrected p-value or a false discovery rate of less than 0.05 to identify the significantly altered metabolites. We found that we had 2,235 ions that were significantly altered. Out of these 2,000 ions, 400 metabolites were found to be significantly altered using univariate analysis. Now we combined the results from the multivariate analysis and the univariate analysis to identify only the metabolites that were significantly altered in both types of analysis. So we end up with 131 metabolites that were altered between the groups based on multivariate analysis and univariate analysis. And these metabolites that were subjected for the pathway analysis. In order to see how is phospholipid gold nanorods are affecting the metabolome of the MCF7 in comparison to the big gold nanorods, we did what we call it volcano plot. Now, as you can see in the volcano plot, colored metabolites or colored circles refers to the ions or the for the ions or the metabolites that were significantly altered and showed a fold change higher than 1.5 or less than 0.5. So we specified that the level of the fold change. So all colored, whether with red or blue, they represent ions or metabolites that were significantly altered and showed a change in their level upon treatment with higher than 1.5 or less than 0.5. Red circles refers to upregulation, while the blue color refers to downregulation metabolites or ions. Obviously, as you can see, this is the volcano plot for the phospholipid gold nanorods treated MCF7s, while this one was the effect of big gold nanorods in comparison to control. In both cases, we are comparing to untreated control. Obviously, you can see that the phospholipid gold nanorods induced a much more significant and a higher impact on the level of metabolome in MCF7 compared to the pig. So it altered a larger number of metabolites, while in the pig, we end up with only a small number of metabolites to be changed. And this might also explain why phospholipid gold nanorods are associated with a higher cytotoxic effect than the pig. It affected larger number of metabolites in MCF7s if we want to compare it with pig. Heat map analysis also showed that phospholipid gold nanorods were associated with a more obvious change in the level of metabolites. This is the heat map. Again, red color refers to upregulation, green color refers to downregulation or decrease in the level. These are the blue color. Can you see the cursor? The blue, yes?
Yes, it's clear. It's clear.
Okay, okay. So the dark blue color here refers to the 10 replicates of MCF7s treated with PEG, while these are the other colors here refer to the control and PEG treated gold nanorods. As you can see that the phospholipid gold nanorods induced a more significant effect, an obvious effect and drastic effect on so many metabolites, while PEG was more similar. Actually, the effect of the PEG gold nanorods was not that significant, and it was almost similar to a control in the heat map, which is consistent with our previous results that phospholipid gold nanorods were associated with higher cytotoxic effect. Interestingly, among the metabolites that were significantly altered in MCF7 cells was choline and taurine. These two metabolites actually are known biomarkers for apoptosis in cancer cells. As you can see that the level of choline and taurine both was significantly decreased in phospholipid treated MCF7, which might indicate that phospholipid gold nanorods indicate their produce their cytotoxic effect through apoptosis mechanism. Now, what we done after that, so it's obvious why now phospholipid gold nanorods are associated with higher cytotoxicity. They are affecting the metabolome of MCF7 to a larger impact. They are affecting larger number of metabolites if we want to compare it with the big. So what we did after that, now we want to know the exact mechanism, how phospholipid gold nanorods are affecting the MCF7s, why it's introducing this cytotoxic effect. What are the underlying biochemical pathways altered? So what we did, we took these 131 metabolites that were significantly altered in both analyses, and we subjected these 131 metabolites to pathway analysis. And I tried to put all the altered pathways in a one figure. So as you can see from the figure, so many biochemical processes were altered in MCF7s upon treating the cells with phospholipid gold nanorods. For example, if we can see here that phospholipid gold nanorods significantly altered the energy metabolism pathways. So first of all, MCF7 phospholipid gold nanorods induce a dysregulation in the TCA cycle, which is responsible for oxidative phosphorylation and producing ATP. And this was indicated by the significant decrease in the level of 4 to 5 intermediates involved in TCA. We also know that cancer cells not only rely on TCA cycle for the production of ATP, they rely on also glycolysis process in order to provide ATP. And as we all know, cancer cells need ATP and they need energy and ATP in order to proliferate in a very high rate. And for metastasis and their growth, they highly rely on the energy pathways to provide a high proliferation rate. So TCA cycle was dysregulated, shut down, almost decreased. The glycolysis pathway was also inhibited or dysregulated, significant decrease, as you can see. Okay, the third arm for the energy production in cancer cells is for fatty acid oxidation. So again, we noticed that so many metabolites involved in the fatty acid oxidation regulated, which means that phospholipid gold nanorods induced energy crisis in MCF7. It almost shut down most of the energy pathways required to produce ATP, which is needed for the proliferation of these cancer cells. So it's inhibited the production of the energy. Also, among the significantly altered pathways, we noticed that phospholipid gold nanorods highly affected the nucleotide metabolism. It affected both purine synthesis and pyrimidine synthesis, which are highly needed for DNA replication and the production of RNA and consequently proliferation. So also this affected the proliferation of cancer cell and might be related to its effect on the cytotoxicity, on their cytotoxic effect on MCF7. Also, among the significantly altered pathways, we noticed that phospholipid gold nanorods altered the cysteine metabolism and affected the redox homeostasis, as indicated by their effect on the NADH and NADPH, and also its effect on oxidized and reduced glutathione. So imbalance, we noticed imbalance in the redox homeostasis. The last path, or the most significant pathways that was altered is mainly related to the amino acid metabolism. And as you can see, so many amino acids were altered, but among them, the most important one was arginine metabolism and glutamine metabolisms, which are highly needed for the cell proliferation. So for example, ornithine was found to be downregulated, and ornithine is highly needed for the production of polyamines, which is required for the metastasis of the cancer cell. So this might also explain the high cytotoxicity effect of phospholipid gold nanorods in comparison to PEG. Now, all these biochemical pathways that were significantly altered with phospholipid are highly needed for the proliferation and the growth of the cell. That's why phospholipid was associated with high toxicity. It's shut down most of these pathways. When we want to compare it with PEG, we notice that PEG, treating the MCF7 cells with PEG gold nanorods only affected to a certain level glycolysis pathway and the pyrimidine synthesis. It did not affect TCA or the fatty acids or the amino acids. And this can also explain the lower cytotoxic effect of MCF7's, big gold nanorods on MCF7 in comparison to the phospholipid. Now, in conclusion, we can say that metabolomics data revealed that phospholipid gold nanorods resulted in a more significant impact in the cellular biochemical pathways than PIG due to their surface chemistry. It's obvious that we had a larger uptake of the phospholipid gold nanorods since it has a phospholipid modification. So it will have a higher uptake. We noticed that phospholipid gold nanorods induced perturbation in metabolism pathways essential for the growth and the proliferation of cancer cells. And this included dysregulation in the TCA cycle, downregulation on the glycolysis process, imbalance in the redox state and the redox homeostasis, impairment in the nucleotide metabolism, and altering the amino acid pool were also noticed. The effect of PIG, as I said, was only limited to the glycolysis pathway and the pyrimidine metabolism. And the most important thing in the conclusion is to highlight the importance of metabolomic as a valuable analytical technique to explore the mechanisms and the cytotoxicity effect of different gold nanorods or nanoparticles in general based on their surface chemistry. As you can notice that gold nanoparticles affected so many or targets or hit so many biochemical pathways in MCF7 cells, and this highlighted the importance of metabolic modulation as a new therapy or a new target in developing therapeutics for cancer, which is metabolic manipulation. At the end, I want to thank the Deanship of Scientific Research at the University of Jordan and Zaytuna University for funding this research. I want also to thank the Center of Analytical Biosciences at the University of Nottingham for running the samples on LCMS and providing us with the raw data so we can do the analysis here on. Thank you so much for your listening, and thank you for listening, and I'm so happy to answer any questions. Thank you.
Thank you, Dr. Lina. Thank you for this valuable presentation with all this information, and you clarify many, many ideas. Thank you very much. As usual, you are brilliant. I don't know, maybe something like, for me, for my background as a pharmaceutics person, what we found that phospholipids are more significant, in affecting all the pathway that you search. So this is due to the nature of this lipid, do you think?
Yes. We did after that uptake experiments, and we noticed that the level of gold nanorods, phospholipid gold nanorods inside the cell is much higher than the big. So the structural modification enhanced the uptake of this gold nanoparticle inside the cells. But how it affects the cell? What are the biochemical pathways that altered might be also explained through the metabolomics. So it might be one of the reasons that its uptake was much higher, but again, it's affected much more pathways in comparison to the big.
Okay, you talk about software that you use to, which helps you to analyze your data. Can you tell us briefly about this software, or maybe something that you use in your research, in this research?
Yes, so for analysis generally in big data, which is known in omics or metabolomics, we use what we call it multivariate statistical analysis softwares, such as the SIMCA, which is a well-known one. And another one is Metabo Analyst. So Metabo Analyst, SIMCA is not freely available. So you have to buy the license for SIMCA. And to be clear, these are softwares for big data. So they are not only used for metabolomics. You can use them also for examples for IR microspectroscopy when you end up with big data. SIMCA is not, as I said, SIMCA is not a freely available software or platform for statistical analysis, while Metabo Analyst is freely available. And actually you can do both univariate analysis and multivariate analysis using Metabo Analyst. I'm not sure if Metabo, just if I can share again, if some of the audience are postgraduates and they are actually interested in seeing or in analyzing the data. So
Yes.
I'm sharing my screen again. So this is Metabo. Can you see now the?
Yes,
yes.
you can.
So this is Metabo Analyst. As I said, it's completely freely available software. It's always this software is always updated. So you can click here to start. You can do so many types of analysis. As I said, you can do univariate, which I mean by univariate, you can do ANOVA, corrected p-value t-test, folded change, volcano plots, heat map. All of the figures that you've seen in my presentation were generated, most of them using this software or a platform. It is, again, it is specialized in large data, big data. When you click here to start, you can see that there is a large number of options that you can use. So you can use it for biomarker analysis, statistical analysis, enrichment analysis, pathway analysis. So many applications. It depends on what is your experiment and what you want to see. So I use a statistical analysis where you can obtain the multivariate analysis and the univariate analysis.
Great. There's a question from a friend. He sent me this a question. Say, you mentioned to use metabolomics to monitor the effect of metformin in healthy subjects. So what was the aim of the study and why you use this healthy subject?
So in metformin's experiment, we used a healthy subject. Actually, metformin is well known now that it can target so many diseases. So it's not only for diabetes. So it can be used for, it's now used in polycystic ovarian syndrome. It is used in cancer as adjunctive therapy. So it has pleiotropic mechanisms. It hits so many targets. But the exact mechanism, how metformin is working out and why it is involved in so many pathways was not clear. So we used metabolomics, since metabolomics study provided profiling to see everything in order to identify the pathways or the targets that can help us to know how metformin is exerting its effect. And we used actually healthy subjects, since when you use a diseased patients, in this case, you can't actually know. It will be the experimental design should be very clear, because when you want to monitor the effect of the drug in diseased patient, you don't know if this effect is related to the disease itself or it's related to the drug. So when you use healthy subject, it's much clear that when you see an effect, this is related to the metformin hitting this target.
Yeah. Thank you very much. Thank you. I don't know if there's any question from the audience.
Would you please, Dr. Masar?
Yes, Dr. Go ahead.
Thank you so much, Dr. Lina. Really very nice, interesting presentation. I want just to comment that one of my MSc students have done the metabolomic studies, and he will going to display the work during the next hour. The most interesting thing that we didn't use the Simca software because it was very costly. We asked for it. They asked about $4,000 to have the access to this software. So we did all the calculations and drawing the graphs by our own, and you will see it. It was very nice. I want to ask you about what do you think about the inter-individual variations and how it could affect the results of such studies?
Yes. One of the main important things in metabolomics, especially if you are working with patients or subjects, is the study design. So the study design is very important. The inclusion criteria, if you are talking about subjects, the inclusion criteria, the exclusion criteria should be very clear from the beginning. So the most important thing to minimize the covariables and the variables. So having matched subjects between the compared groups should be, this is highly needed. So similar age, body mass index, any demographic, other data should be matched. You will try your best to match the two groups. But at the end, as you said, there is always variability between subjects. And that's why most of the time when with biological samples, we don't see clear separation using the PCA plot. We used the OPLSDL plot. But the only thing that you can do is to try to make them match as possible and specify from the beginning a strict inclusion and exclusion criteria. So the only difference between the groups might be mainly related to the condition that you are studying. And I just want to let you know that now, instead of drawing the figures in Metabolomics, you can use Metabo Analyst. It's a freely available, and the last version of Metabo Analyst couldn't identify the significantly altered metabolites in OPLSDA plots or in multivariate. But nowadays, the last version, the one that I showed you just in a minute, it can do both multi and unique,
Yes.
which means that you don't need Simca now.
Oh, okay. Okay. Thank you.
Thank you, Dr. Anham. I don't know if there is someone want to ask Dr. Lina, because really it's interesting what you do. It's a great work. I think we don't have anyone to ask because you clarify everything actually. Thank you, Dr. Lina. Thank you for let us see you this night. And thank you very much and hope you the best. Thank you very much. Dear audience, now we'll move to Dr. Zakaria and Dr. Ara to go to our oral presentation that are included in this day. Dr. Zakaria, Dr. Ara, are you ready?
Hello. Thank you, Dr. Massa.
The floor is yours now. Thank you very much.
Thank you. Thank you. Now we complete the conference by starting oral presentation session. The first presentation will be introduced by the pharmacist Samer Hassan Ahmed, who currently works in Mustansiriyah University College of Pharmacy, Department of Pharmacology and Toxicology. And his research title is Hepatic Metabolomic Profile of Atorvastatin and Fenofibrate in Hyperlipidemic Rats. Welcome, Dr. Samer, and you can now share your presentation, please. Dr. Samer.
Yes, good evening, Dr. Zakaria. Thank you. I am now sharing my slide.
You may go ahead. Everything is clear, Dr. Samer.
Yes, today I will present hepatic metabolomic profile of atorvastatin and fenofibrate in hyperlipidemic rats. I will introduce from now about metabolomic in hyperlipidemic research, aim of study, methodology, result, and finally conclusion. hyperlipidemia in the initial state happened mainly in individuals who have no symptoms and therefore sometimes misdiagnosed. The structure existent of hyperlipidemia make modification to the endogenous metabolite, and this may have a prognostic index at the early stage of this condition compared with the level of lipid at asymptomatic phase. One of the core challenge doctor and research face surrounding hyperlipidemia is early diagnosis. Metabolomics method provide further information about altered pathway and disease causes and consequences, and therefore metabolomics play an important part in understanding the effect of metabolomic for prognostic purpose. Atorvastatin and fenofibrate both are most commonly chosen drug for treatment of hyperlipidemia as a mono or combination therapy, despite the fact that pharmacological mechanism extensively studied, but their efficacy and side effect in patients with hyperlipidemia throughout long-term treatment seem confused. As hyperlipidemia is a systemic disease that affects the metabolism of many endogenous small molecular metabolites, therefore understanding the interference of atorvastatin or fenofibrate inside the body metabolic system is essential. Aim of the study is to investigate the metabolic map using GC-mass-based metabolomic approach to assess lipid regulating effect and adverse reaction of atorvastatin, fenofibrate, and combination in diet-induced hyperlipidemic rat, and looking for a new biomarker for hyperlipidemia using a GC-mass and assessing the lipid profile. 30 male adult rats allocated to 5 groups at random, each with 6 rats, normal, model control, and treated group. High cholesterol diet was prepared by mixing 2% weight per weight cholesterol and 28% weight per weight beef tallow and 70% weight per weight normal diet powder. At the end of experiment, at 8 o'clock, 2 portions of blood was collected, and then serum and plasma was obtained, used for lipid profile analysis and metabolomic analysis, respectively. Metabolomic workflow. First, sample preparation was done by protein remove and metabolite extraction. Sample derivatization done by MSTFA plus 1% trimethylchlorsilane, and data acquisition done by GC-mass. Data analysis was done by ANOVA and principal component analysis. Biomarker identification was done through database such as KEGG and Human Metabolome database. Finally, biomarker interpretation also done by Human Metabolomic database. Changes in plasma lipid parameter in normal model and control group. We can see model control group or lipid parameter significantly increased compared with the normal control group, and this is indicate the success of a hyperlipidemic model. And with the treatment group, we can see all lipid parameter return to the normal level, and the reduction was higher in case of combination due to synergistic effect. Principal component score plot, we can see rat in normal control group assembled in the down right quadrant of the plot, and this is suggested a stable state of metabolism, while rat in model control group assembled on the up and down quadrant of the plot. This is suggested that successful of hyperlipidemic model, and also rat show a different metabolic state from a normal control. And also the rat in the treatment group assembled between normal and model control group. This is indicated that a rat show a different metabolic response to different drug. Variable accountable for classification in principal component analysis score plot are described in BCA loading plot, and we can see here. Sorry. Now, changes in metabolic pathway and metabolite related to carbohydrate in model control group comparing to normal group. Here we can see glucose, galactose, pyruvate significantly increase. This can be give indication that glycolysis inhibition and energy metabolism dysfunction. After the treatment, they return to the normal, and therefore galactose and glucose can be suggested a biomarker for hyperlipidemia. Metabolite related to amino acid, we can see all amino acid decrease
Dr. Samar,
significantly in
sorry
model
for the
control.
interruption. We are not able
Yes.
to see your image.
My slide or my image?
Your
Can you
slide,
share
your slide.
Your slide, please, Dr. Samar. Can you reshare it?
Okay. Now,
Yeah, everything is good now. Maybe if you zoom it a little bit from the 70% down below, increase the percentage.
yes.
And you may wish to hide the toolbar from the top so that you'll be able to have more space.
Okay.
There you go.
Okay. Metabolite related to amino acid metabolism, where we can see in model control group, all amino acids significantly decrease except for ornithine, which is the intermediate amino acid metabolite in urea cycle. And after the treatment, ornithine it's returned to normal level, and this can be suggested as a biomarker for hyperlipidemia. Also, phenylalanine and its amino acid play important role in lipid metabolism through tyrosine epinephrine pathway. It's appear only in normal control group and disappear in model control group and can be suggested as a biomarker for hyperlipidemia. On the other hand, a metabolite related to lipid metabolism in model control group, which here in red arrow, we can see all saturated fatty acids significantly increase, while unsaturated fatty acid, polyunsaturated fatty acid, which is beneficial, disappear in model control group. After the treatment, saturated fatty acid decrease and polyunsaturated fatty acid increase in all treated group. And also this can be suggested a biomarker for hyperlipidemia. Cholesterol appear only in model control group. Metabolite and metabolic pathway related to ketone body formation due to high amount of acetyl-CoA, one portion generated from beta oxidation and another portion generated from amino acid metabolism by gluconeogenesis, and another portion of acetyl-CoA come from glucose inhibition due to high cholesterol level in the body, so it inhibits endogenous cholesterol synthesis and lead to accumulation of amount of acetyl-CoA. All these acetyl-CoA can generated into ketone bodies such as acetoacetate, acetate, and 3-hydroxybutyric acid. And the level of this metabolite significant increase in model control and decrease in case of a treatment group or disappear. And also this can be suggested as a biomarker for hyperlipidemia. To conclude, a metabolic change in model control group relative to normal control group, we can see all amino acids significantly decrease except for ornithine. Also, carbohydrate significantly increased. Cholesterol appear only in model control group. Polysaturated fatty acid, saturated fatty acid appear significantly increased, and beneficial polyunsaturated fatty acid appear only in normal control group. Metabolomic change in the treated group relative to model control group, we can see ornithine significantly decrease on all treated group. Carbohydrates significantly decrease. Cholesterol, as mentioned before, appear only in model control group. Saturated fatty acid significantly decrease. Polyunsaturated fatty acid appear only in normal control group and in treated group. So to conclude, according to the result of this study, it was confirmed that high cholesterol diet significantly induced hyperlipidemia, and metabolic profile approach based on GC-mass assay has been successfully applied to monitoring pathological changes and therapeutic process associated with atorvastatin, fenofibrate, and combination treatment in high cholesterol diet-induced hyperlipidemia. The present study identifies a new series of potential biomarkers by global metabolomics and may be used to explain a metabolic effect of hyperlipidemia during disease progression and recovery effect of lipid-lowering agent, including acetoacetate and 3-hydroxybutyric acid, linoleic acid, oranthine, phenylalanine, galactose and D-glucose, and pyruvic acid. Thank you for your attention and for your patience.
Thank you, Dr. Bassam. Thank you for your nice presentation. Dear audience, if you have any question, it will be answered at the end of the session by the presenter. And now the second presentation will be introduced by the pharmacist Lina Qasim, who currently works in the Mustansiriyah University College of Pharmacy, Department of Pharmacology and Toxicology. And her research title is Evaluation of Renoprotective Effect of lipoic acid and bosentan against diclofenac-induced acute renal failure. Welcome, Dr. Lina. Your mic, please. We can't hear it, Dr. Lina.
Dr. Lina, maybe if you take out the headphones and try again using the regular laptop speaker.
You hear us, Dr. Lina?
She can hear us, but we can't hear her.
Oh.
You need to go to the bottom left and select a microphone from the bottom of the screen at the toolbar. No, you're not audible. Perhaps if you log out and get back again. Dr. Zakaria, maybe if you move on to the next presenter, please.
Okay. And the next presenter will be Dr. Haidar Tamimi, who currently works in Mustansiriyah University College of Pharmacy. And his research title is Molecular Docking Synthesis, Characterization, and Antiproliferative Evaluation by Pyrozoline Derivatives. Welcome, Dr. Haidar. Can you hear me?
Welcome, Dr. Zakaria. Yes, I can hear you.
Welcome, Doctor. You can share your presentation, please? And you have 10 minutes for presentation as a reminder.
Okay. Hello, everyone. My name is Haidar. Today, I will present my research work, which was about breast cancer. The title of my project is Molecular Docking Synthesis, Characterization, and Antiproliferative Evaluation of Pyrozoline Derivatives. The outline, introduction, the aim of the study, method to conduct our target, the results, conclusion. We first start with introduction. Background: breast cancer is one of the most prevalent malignancy among women across the world. Breast cancer is diagnosed in over 1.5 million women worldwide each year. It's about 25% of all women with cancer. Breast cancer is divided into 3 subtypes: estrogen receptor positive, progesterone receptor positive, human epidermal growth factor receptor 2, which abbreviated by HER2 positive. Therapeutic choice: chemotherapy, hormonal therapy like tamoxifen, or an aromatase inhibitor like letrozole, HER2 drug target like trastuzumab, surgery and radiation. The lack of estrogen receptor positive, progesterone receptor positive, HER2 positive receptor resulted in discovery triple negative breast cancer. It's aggressive and few therapeutic choice. So why do we choose pyrazoline ring as a scaffold? Pyrazoline constitutes a principal heterocyclic family containing 2 nitrogen atoms in their 5-member ring, exhibiting a wide range of chemical, biological, and pharmacological properties. Pyrazoline is a basic in nature, has only one double bond between the N2 and C3, one double bond only. Pyrazoline and sulfonamide-derived compound have gained extensive research attention due to their cytotoxicity and antiproliferative activity. The aim of the study: molecular docking on both target human estrogen receptor and poly-ATP ribose polymer enzyme 1, PARP1, using gold suit. Synthesis a new series of pyrazoline derivatives. Characterization and in vitro antiproliferative study on both target estrogen receptor positive and estrogen receptor negative cell line. In silico ADME study using Swiss ADME suit. Method to conduct our target. We follow up scheme of chemical synthesis, start with acetophenone and different aldehyde derivatives under ultrasonic radiation to give chalcone, then cyclization reaction to give pyrazoline derivatives. Instrument used to confirm compound: FTIR, HNMR, CNMR. Computational method: measure of drug likeness, system preparation, energy minimization, molecular and ensemble docking, binding energy calculation, PLP fitness score. The result: the binding energy of pyrazoline derivatives and tamoxifen using gold suit or gold program on a human estrogen receptor. We found the compound 10 give the best score, 98, while the tamoxifen give 94 only. The binding energy of pyrazoline derivative on PARP1, we found that compound 8 give the highest score, 92.03, is the best of all final compound. This figure represents a 3D dimensional binding mode of compound 10 on a human estrogen receptor. The green color represents the hydrogen bond, while the red color represents the other bond on this target. Figure 2 represents the 3D dimensional binding mode on PARP1 target. Also green color hydrogen bond and red color is another bond. Table 3 represents pharmacokinetic properties of synthesized compound using Swiss ADME suit. We found all compound is high GIT absorption with zero Lipinski violation. The FTIR spectrum of compound 10, we can remark the 3 main characteristic peak, one on 5093, which belong to C double bond N, the another 2, 3392 and 3273, which belong to NH2. The HNMR spectrum of compound, which we can remark the 3 peak. The first one for this H that belong to CH2 of pyrazoline duplet at 3.11 chemical shifts, while the second H, we remark on CH2 pyrozoline doublet 3.87. The third H of pyrozoline ring for CH of pyrozoline doublet at 5.50 chemical shift. The CNMR spectrum of compound 10, we remark also three main peaks, one for CH2 of pyrozoline at 43, the second one CH of pyrozoline ring at 62.54, the third main peak which belong to C double bond N at 150. The anti-proliferative activity on post cell line, we found that compound 10 is the best score, about 4.6 micromolar on MCF cell line, which represent the human estrogen positive receptor, while the compound 8 give 7.70 on MDA-MP cell line, which represent the estrogen receptor negative. The conclusion, a successful chemical synthesis of pyrozoline derivatives linked with sulfonamide moiety. ARM studies show that all design compounds highly absorb from the GIT. The anti-proliferative evaluation synthesized compounds show an excellent promoting anti-breast cancer activity, especially the compound 10 that evaluated on MDA-MP 468 cell line that represent the human the estrogen receptor negative, while the excellent activity of compound 10 on MCF cell line than tamoxifen. This is the reference, and thank you very much.
Thank you, Dr. Haidar, for your nice presentation and valuable work. Thank you.
Thank you very much. Thank you very much.
Dr. Lina, if you hear me.
Yes, doctor, I hear you. Do you hear me?
Okay.
Okay.
Welcome back. And
thanks.
You can now share your presentation.
Okay.
You have 10 minutes for your presentation. Thank you. Go ahead.
Okay. Good evening. My research subject is about evaluation of the venoprotective effect of lipoic acid and prosentan against diclofenac-induced acute renal failure, supervised by Prof. Dr. Ghaith Ali Jassim, Assistant Prof. Dr. Ihsan Salah Muhammad Bida. Acute renal failure, a recent term, is acute kidney injury, is a syndrome in which the glomerular filtration rate declines abruptly within hours to days, and it is usually reversible. Approximately 19 to 33% of acute kidney injury episodes in hospitalized patients are related to the drug-induced nephrotoxicity. There are many causes for acute renal failure. We have first the prerenal cause, which is about 55%, including volume debilitation, low cardiac output, increase the vascular resistance, like in our case, non-steroid and anti-inflammatory drug use. Second, we have the 40%, which is around 40%, which is the intrinsic cause, like we have the acute tubular necrosis and acute interstitial nephritis. Third, we have the post-renal cause, which is around 5%, like in urinary flow obstruction, which is acute in the prostatic gland disorder, and the large renal pelvis stone. Diclofenac-induced acute renal failure. Although it's considered safe, but the diclofenac have gained special attention in the past few years due to the potential risk of the renal damage, in which non-steroidal can cause two different types of the acute kidney injury. First, we have the hemodynamically mediated, and second, the immunomediated. Pathophysiology of the acute renal failure induced by diclofenac. Non-steroidal, the diclofenac has direct effect on kidney by causing mitochondrial dysfunction, which is resulting in the formation of active oxygen species and active nitrogen species formation and oxidative stress. Also, non-steroidal cause decrease in the prostaglandin synthesis by inhibiting the COX enzyme, resulting in afferent arterial vasoconstriction that leading to the decrease of the renal blood flow and oxygen delivery, resulting in renal ischemia, which has a microvascular effect by causing the endothelial dysfunction, which is increase the endothelin-1 release and decrease the vasodilatation response to nitric oxide and prostacyclin, increase the vasoconstriction response to the endothelin-1, angiotensin, and adenosine, and increase the leukocyte endothelial adhesion inflammation. Also, renal ischemia has a tubular effect by causing the cytoskeleton breakdown, loss of polarity, and so on, resulting in the tubular obstruction finally. The aim of our study is investigating the effect of lipoic acid and porcentane in diclofenac-induced acute renal failure in the male rats. The lipoic acid, also called the thiotic acid, has antioxidant activity by acting as a free radical scavenger of the active oxygen species and the active nitrogen species, also acting as a metal chelator for the copper and the ferrous, and regenerator of endogenous antioxidants, such as vitamin E and vitamin C. And also lipoic acid has anti-inflammatory activities by inhibiting nuclear factor Gabapentine, which is resulting in decrease the pro-inflammatory mediator, and also inhibited the vascular cell adhesion molecule 1, intercellular adhesion molecule 2 production. And finally, it's also activated the endothelial nitric oxide synthase through phosphosidine citrate triphosphate kinase acid signaling pathway. Porcentane is considered a competitive antagonist for endothelin-1 at 2 receptors, A and B. When endothelin-1 binding to its A receptor, it's cause afferent arterial vasoconstriction, increase from the pro-inflammatory mediator, and increase and upregulated the expression of adhesion molecule on vascular endothelial cells. And this induced the aggregation of the polymorphonuclear neutrophils, induced the synthesis of extracellular matrix components such as fibronectin. And on the other side, when it's binding to its B receptor, it's activated the prostacyclin synthase, nitric oxide synthase, leading to the formation of the nitric oxide and prostacyclin, which is resulting in a vasodilator effect. The methodology here, we took 30 adult male albino rat, which is weighing around 200 to the 250 gram, which is divided to the 5 group: control, induction, lipoic acid, porcentane, and combination. For the first 5 days, both of control and induction taking distilled water 5 ml per kg by mouth, while the lipoic acid received 200 milligram per kg and porcentane 100 milligram per kg, and combination group received 100 milligram for both of the lipoic acid and porcentane. Then at the 5th day, control group receive normal saline intraperitoneal, while the rest of the group induced acute renal failure by diclofenac sodium by through administration 100 milligram per kg intraperitoneal. Then for the other 5 days, we follow the same the protocol treatment that we follow in the first 5 days. The biomarkers measures in our study, here we have serum urea and the serum creatinine, which is measured by the calorimetric technique. We have also serum superoxide dismutase, malondialdehyde, fibronectin, transforming growth factors beta-1, and serum kidney injury molecule 1, which is measured by the sandwich enzyme linked immunosorbent assay technique. Our result here, as we see here, on the left side, we see there is increase the mean serum creatinine level in induction group were significantly elevated in compared to control group, while there is no significant differences in the lipoic and porcentane in comparison to induction. And in the combination group, there is a significant decrease in the mean serum creatinine level in compared to induction group. The same result here we're getting on the right side according to the urea, in which this could be because of lipoic acid act as antioxidant, so decrease from the oxidative stress and decreasing in turn the overexpression of endothelin-1, and because also the porcentane block the action of endothelin, so this will be inhibited the afferent arterial vasoconstriction. This will be improve the glomerular filtration rate and decrease the serum creatinine and urea level. Also, we have the effect of the lipoic acid ampocentan and combination therapy on the oxidative stress. On the left side, we have the malondialdehyde, which is significantly elevated in induction group and compared to control group. And there is also no significant difference in the lipoic and porcentanic group in compared to induction, while the mean serum malondialdehyde level in the combination group will be significantly decreased in compared to induction group. This could be because, on other side, the superoxide dismutase significantly decreased in induction group and compared to control, and there is no significant difference here in the lipoic and porcentanic group and compared to induction, while there is significant elevated of the superoxide dismutase in the combination compared to the rest of the group. This could be because of lipoic acid approved to decrease the NADPH oxidase 2 and NADPH oxidase 4, which is a major source for the reactive oxygen species, so decrease from the oxidative stress, and the porcentan also approved by previous study increase the endothionatic oxide synthase through the phosphosidine and triphosphate activation. So this will be also decrease the oxidative stress, leading to the malondialdehyde serum level to be decreased and superoxide increase. The effect also on the kidney injury molecule 1, there is a significant increase in the induction group comparing to control, and there is no significant differences between the lipoic and porcentan comparing to induction group, while there is a significant decrease in the combination group when we're comparing it to the induction group and the lipoic acid and porcentan. This could be because both of lipoic acid and porcentan approved before, according to the previous study, that will play an important role to inhibit the phosphorylation of the P38 mitogen activated protein kinase pathway, which is a play important role to shedding the KIM1 from the proximal convoluted tubule. So this leading to decrease the KIM1 release. Also, we having here the effect of the treatment on the transforming growth factor beta and the fibronectin. They're giving the similar result in which there is significant increase in transforming growth factor and fibronectin in the induction group in comparing to control, while there is no significant difference in the lipoic and porcentanic group in comparing to the induction and control. Here, with the combination group, shows a significant decrease in the transforming growth factor beta and fibronectin in the combination comparing to the induction group and the lipoic acid, but there is no significant differences with the lipoic acid and porcentan here. Also, this could be, as I said before, this may be due to the inhibition phosphorylation, which is also play important role the P38 mitogen activated protein kinase pathway to releasing of transforming growth factor beta and fibronectin aggregation. So this maybe could be the reason behind this decrease. By applying the person correlations analysis, we're getting here a moderated positive relationship between the KIM1 and urea and between KIM1 and creatinine. Also, there is a relationship considered significant but weak between fibronectin and creatinine and fibronectin with urea. But interestingly, there is a moderated positive relationship between KIM1 and fibronectin. And then we apply the received and receiving operating characteristic analysis by applying this row curve for the KIM1 biomarkers. We're getting the area under the curve is around 0.986, which is considered as very good value. Also, we applied here the row curve for the fibronectin biomarker. We're getting the area under the curve about 0.829, which is considered a good value. So we conclude from our study the pre-treatment with the combination therapy of lipoic acid and porcentan prevent the renal damage significantly in the rat model of diclofenac-induced acute kidney injury. Also, fibronectin is considered a promising biomarker for detection and diagnosis of acute kidney injury. Thank you very much for listening.
Thank you, Dr. Lina, for the awesome presentation. Thank you very much.
Thank you, Doctor.
And the next presentation will be introduced by the pharmacist Haider, by Muhammad Yawuz Jamal, who currently works in University of Baghdad College of Pharmacy. And The research title is Clinical Significance of Brussels Anti-Adalimumab Antibodies in Patients Serum with Rheumatoid Arthritis. Welcome, Dr. Muhammad. Can you hear me, Dr. Muhammad?
Yes, I can hear you, Doctor. Thank you very much. Good evening, everyone.
Thank you. You have 10 minutes for your presentation. Good luck.
Yes,
and
thank you.
Lina, you can stop your presentation. Okay, thank you.
Our research about the clinical significance of the presence anti-adalimumab antibodies in patients serum with RA, done by researcher Muhammad Yawaz and under supervision Nazar Abdul Latif and Dr. Nasir Al-Harjan. بسم الله الرحمن الرحيم وما أوتيتم من العلم إلا قليلا صدق الله العظيم. RA is the chronic systemic autoimmune inflammatory disease which is primarily affect the joint and is characterized by inflammation of synovial joint in symmetrical pattern. RA can also produce extra articular manifestations and diffuse inflammation in the lung, pleural, pericardium, and sclera. Also nodular lesion in the subcutaneous tissue. Epidemiology, approximately 1% of the population worldwide is affected by rheumatoid arthritis. نسبة ليست قليلة 1%, with female being 2 to 3 times more commonly affected. Prevalence RA increase with age in both gender, nearly 5% of women and 3% of men over age 65 were affected by disease. As we show in the global prevalence rate of RA in the worldwide, we see Iraq in the circle, red circle, is prevalence about 1.4 to 1.5% in compared to other region in Asia. We see the high prevalence rate in Iraq. Etiology and risk factors. There are genetic factors and environmental trigger. A genetic factor play a role in the development RA as well as in the pathophysiology of disease. Environmental factor, smoking, a hormonal factor, bacterial, viruses, and diet. In pathogenesis, in healthy individual, there is balance between pro-inflammatory cytokine, who interleukin 1 and 6, with TNF-alpha, with anti-inflammatory cytokines, example, interleukin 1 receptor antagonist, interleukin 4, and 10 interleukin 11. In synovium of patients with RA, however, this balance between pro-inflammatory and inflammatory cytokines is disrupted. Thereby, towards to the pro-inflammatory cytokine, as we say, interleukin 1, 6, and TNF-alpha. This diagram shows the pathogenesis when the antigen is elicit T cell and then B cell introduce antibodies with complement to induce the macrophage to release TNF. These are some site of drug interaction with etanercept and adalimumab. This is our drugs, adalimumab, interact with TNF, and this is site of the methotrexate and leflunomide. American Rheumatism Association criteria for diagnosis of rheumatoid arthritis: well, morning stiffness, swelling of 3 or more joints for at least 6 weeks, symmetric joint swelling for 6 weeks, hand X-ray changes, rheumatoid subcutaneous nodules, positive RF. How we can assess the patient with RA? We can by using a disease activity score, 28 ES with ESR. This can be calculated by using application of the mobile. A simple can use can divide the patient to the mild, moderate, and severe RA. 5.1 and more, this is severe. From 5 to 1 to 3 to 0.2 is moderate, and less than 3 is low disease activity. And treatment, there are 4 classes of medication in RA: non-steroidal, or corticosteroid, or disease-modifying anti-rheumatic disease drugs, or biological agents. This is biological agent, a TNF blocker. We have the chimeric, humanized and fully human. Is our drugs is adalimumab, where this is the humanized receptors. This is our drugs, adalimumab, fully humanized. Adalimumab Humira is genetically engineered, fully humanized IgG1 monoclonal antibody, is self-administered subcut by patient as 40 milligram injection every other weeks, has terminal half-life comparable to weeks, long half-life, can be take patient one injection twice weekly. Patient method study design, prospective observational cohort study. During of study is 9 months, follow up 60 patient, established with and diagnosed with RA, receiving biological agent with adalimumab, Humira, with or without conventional synthetic disease modifying drug, DMARD. Exclusion criteria, inclusion criteria. Exclusion criteria: active infection, hepatitis, TB, heart failure class 3 and 4. Inclusion criteria: patient over 18 years, patient with adalimumab treated with rheumatoid arthritis. The baseline characteristic: adult age, mean 45, where disease duration mean كانت 9 years. ونون adalimumab antibodies وكانت المين مالته 7.7 والبودي ماس index 28 and overweight. Other demographic data of the patient: genders, male and female is classified 14 and 46. Smoker and non-smokers. Most of the patients participant in the study was non-smoker. RF factor positive 56, negative 4. Anti-CCP positive 44, negative 16. Will can use DMARD 54. All other biological agent used previously, most of them can that not use a biological agent previously. Compare between parameter RA patient treated with adalimumab baseline and after 3 months of treatment. As we see this ESR, there is no significant difference, while the score does 28 highly significant. HB, there is no change below. HB, YBC, AST, ILT with serum creatinine. El mean does score 28 at baseline after 3 months, as we show in this figure. This mean and the score of DAS 28 before treatment was 6. And after treatment, we see decrease and from 6 to 4.5, and P value can highly significant. This is indicate the effectiveness and of the therapy. Relation between DAS score after 3 months with anti-adalimumab antibody was anti-adalimumab antibody 3 AAA anti-adalimumab antibodies positive was the score marathon كانت 5 high score, while the patient with negative anti-adalimumab antibodies have low DAS score. This is indicate that presence of these antibodies to the drug IgG1 adalimumab, this decreased effectiveness of the treatment. And this is like show the relationship between percent of patient and functional class with positive and negative anti-adalimumab antibody. Functional class of the patient divided from 1 to 3 and 4 for severe cases. Functional class 1 can do his life normally. Class 2, there is some restricted in the normal activity, and class 3, there is major restrictive in the daily activity. We see the percent of patient have the positive anti-adalimumab antibody. Patient have anti-adalimumab antibody. Class 1 C is low, while class 2 functional class and decrease in activity, restricted in daily activity is high, and in the third class we see the major of the patient not respond to treatment and have anti-adalimumab antibody develop high in the functional class 3. In conclusion, adalimumab is effective and relatively safe in treatment RA. about 42% of patients taking adalimumab will develop anti-adalimumab antibody. The presence of these anti-adalimumab antibody in patient serum negatively affect on response, as we see that DAS28 is highly in the group of positive anti-adalimumab antibody. A recommendation: this study can be extended over longer period, and larger sample size can support the result. Serum drug monitoring should be considered in clinical practice during long-term use of biological agent. And thank you for listening and watching. And any question, I will be ready. Thank you, Doctor.
Thank you, Dr. Mohammed. Thank you very much. Thank you for presentation. Nice presentation, and for sharing your valuable work. And now the last presentation will be introduced by pharmacist Falah Hassan Hadi, who currently works in University of Kufa College of Pharmacy. And his research title will be Optimizing the Phosphate Level in Patient Among Hemodialysis Patients by Pharmacist Intervention. Welcome, Dr. Falah.
Thank you, Dr. Zakaria.
Welcome. And present your work. Good luck.
Okay. It's okay.
It's okay. Can you make us
good evening for everyone. My research titled as Optimizing the Phosphate Level Among Hemodialysis Patients by Pharmacist Intervention. Firstly, I will go for introduction. Chronic kidney disease is defined as abnormal kidney function or structure presented as albuminuria and or glomerular filtration rate below 60 ml per minute, consistent for 3 months or more, with implication for health. In advance, CKD lead to a many systemic disorder. One of these is chronic kidney disease bone mineral disorder, CKD-MBD. CKD-MBD is a systemic bone and mineral metabolism disorder characterized by one or a combination of biochemical abnormalities such as calcium, phosphate, parathyroid hormone, vitamin D metabolism or level, and or bone abnormalities such as abnormal bone turnover, mineralization, linear growth, volume, or strength. In CKD-MBD, the most detrimental condition that correlate to the cardiovascular and all-cause morbidity and mortality is hyperphosphatemia. Hyperphosphatemia characterized by abnormally elevated serum phosphate level, and invariably occur among end-stage renal disease, especially patients on continuous hemodialysis. It's becoming more and more substantial clinical entity due to its strong link with morbidity and mortality. In CKD,
sorry for interrupt.
Okay.
I think the slide will be just fixed in the first one.
Sorry, it's maybe internet. It's right now. It's okay now.
The same slide. It's just one.
Okay, maybe internet problem. My voice.
Exit from the. I think you now in the slideshare.
Slideshare, yes, please.
Just exit from the slideshare. I think you have a problem with the.
It's okay now.
Okay now.
Okay now.
It's okay.
Okay, but so it's the hyperphosphatemia mainly its risk mainly due to its potential to cause vascular calcification and stiffness and lead in future to secondary hyperparathyroidism. According to the recent guidelines, such as KDIGO 2017, it's treated by phosphate binder medication, dialysis therapy, and or low phosphate diet approach. So the adherence to the medications in general, it's a complex process affected by a combination of the of many parameters such as socioeconomic parameter and healthcare system parameter, patient attributed parameter, and so on. But the medication adherence specifically is defined as amplitude to which patient behavior match the prescribed drug dosage regimen involving timing, dose, and interval of drug consumption. So non-adherence for phosphate binder is the main issue in this patient population suffering from this condition, which is hyperphosphatemia. So due to many factors, one of the main factors, phosphate binder pills account for more than 50% of daily pill intake of the dialysis patient. Poor adherence toward phosphate binder may be reach up to 99%, according to many variables. But the average value is 53%. Nearly half of patients don't take their therapies as directed by their physician. Medication non-adherence has detrimental impact on safety, efficacy, and cost of treatment. Assessment measures of adherence to phosphate binder are subjective, objective, and biochemical measurement of serum phosphate level. So the subjective one are, which is the most commonly used techniques, including validated and non-validated questionnaires such as MARS-5, BMQ, and so on. Objective measurement, which is minimally used method, such as medication event monitoring system CAPS and medication position ratios, where the last one is biochemical measurement of serum phosphate level. Aims of the study. The current study was attempts to assess the following, assessing the patient adherence and knowledge toward phosphate binder at baseline, وأيضا to see if the pharmacist-led intervention could improve the phosphate binder adherence and lead to the reduction in serum phosphate level. The study was descriptive, interventional, mixed method randomized control study based on validated questionnaire and biochemical parameter conducted in two centers, الصدر Medical Teaching City and Al Hakim General Hospital Nephrology Centers. Has been approved by Scientific and Ethical Committee from the Faculty of Pharmacy, University of Kufa, and by Najaf Research Health Directorate. The study tools, we use three main tools: questionnaire for assessing medication adherence and knowledge at baseline and after intervention. Where the second one, educational material that we interact with patients, such as booklet, calendar, and talk intervention. Talk intervention was a 15-minute continuous counseling specified for each patient by pharmacist about his condition, his phosphate binder used for treating hyperphosphatemia, and the future complication of the hyperphosphatemia, future if not treated well, and the importance of medication adherence, and so on. So this is the Arabic version of 8-age booklet, education for the patient, containing information about phosphate absorption, and high phosphate content, high phosphate content foods, as notified by picture to the patient, and the correct way to take their phosphate binder. For the most commonly used phosphate binder in these two centers was the Sevelamer. So we put the picture of the tablet of Sevelamer and the future complication of Sevelamer, of the hyperphosphatemia and side effect of phosphate binder. This picture for the calendar, this picture for calendar used to be given bi-weekly to the patient in the center to notify the use of phosphate binder dose by himself. The study arms, the study was of 100 dialysis patients recruited from the two center during 2 months, and the net result of the patient, including the study, was 50 patients. that enter and complete the interventional phase completely. Study parameter, medication adherence, medication knowledge, serum phosphate level, and calcium level. So these demographic data to the patient and the risk factor at baseline, this knowledge score, number of pills taken by the patient on daily, phosphate level, calcium level, hemoglobin, albumin levels, which is the nutritional biochemical parameter to the patient. After intervention, as you see, at baseline and after intervention values for the adherence score, knowledge score, phosphate level, calcium level, there is a highly significant improvement in the adherence score and the knowledge score. This highly significant improvement in adherence score and knowledge score lead to the significant improvement in a serum phosphate level and hyperphosphatemia condition in the patients. Conclusion, face-to-face short-term pharmacist educational intervention result in a significant increase in phosphate binder adherence and knowledge score, as well as significant reduction in serum phosphate level and total daily pills, number of pills taken. Reduction recommendation. Another research are needed to assess non-adherence to the dialysis and low phosphate diet. There is also non-adherence to the dialysis and non-adherence to the phosphate to the low phosphate diet approach to treatment of hyperphosphatemia. Or further study involved electronic follow-up system managed by pharmacists in the center to notify patients to take their pills by direct interview and by phone message. Thank you for attendance. Thank you for attendance and watching, and so sorry for taking more than time line.
Thank you, Dr. Fala. Thank you for the presentation and your short word. Thank you for all the presenters for this session. If you hear me, is there any question from the audience to the presenter?
I hear you loud and clear, Dr. Zakaria. Thank you for your handling of the session.
Okay.
By now, before we get into the questions, I'd like to make a quick poll about and take the voting of the audience about the best researchers from yesterday and today. So I'll be trying to post a poll. I hope that it will be shown at your devices. Please let me know if it did. And while it's on, is it on?
Okay, so
it's on, okay. So while we see how the results are being populated, I'll be allowing the questions to pile up again. I believe that Dr. Manar, Dr. Zainab have their questions. By all means, Dr. Zainab, you may go ahead and ask if you wish to.
Okay, my question is to Lina Bahjat.
Yes, doctor.
Yeah, Lina, very nice work and very nice presentation. My question is, what method did you use to measure NDA?
The ELISA sandwich enzyme-linked immunosorbent assay, doctor.
So could you elaborate on this method a little bit?
What, doctor?
How did you measure?
The technique?
Yes.
Yes, I bought the kits from USA by my biosource company. The technique is sandwich. It's called sandwich because there is the antibody attached to the microplate, and we adding the sample which is containing the antigen that will be binding to the antibody that attached to the microplate. Then by adding the other antibody, which is biotinated antibody, that will be interacted with the peroxidase, epidine peroxidase, which is then interacted with the colored product tetramethyl benzidine to giving the color product. Then this color product, we measured it by microplate readers in our college in the lab. Then by taking to the absorbance reading, and we having the standard measuring the sample absorbance and the standard absorbance, and because we have the concentration of the standard and the absorbance also, we can draw the standard curve, and by applying the standard curve, we can get the concentration of the sample by knowing also the absorbance of the sample also. So it's the technique, by applying it, we can get it also according to equation by knowing the absorbance of standard and the concentration of the standard, and by getting the absorbance of the sample, we're getting the concentration of the sample, so we can get the concentration of monodialdehyde in different groups.
Okay, thank you.
Thank you, doctor.
Lina, do you hear me?
Yeah, doctor.
So there's a question to you also. You use a model for acute renal failure, am I right?
Yeah, doctor.
Why did you use this model?
The diclofenac?
You use model for renal failure, like acute failure, not for chronic disease.
Because the time we are restricted in time, so we use the acute renal failure model because we can get and we can induce this model, and by in the futures we can be applied for the chronic. Yeah, because the time restricted here, time restricted, so we attach to the acute renal failure, not the chronic. So the time restricted with time. Yeah.
Okay, thank you, Lina.
Yes, Lina, excuse me. Thank you for the fruitful work. Excuse me, Dr. Zakaria, for just to save time. Thank you for the fruitful results and for all the researchers. Very interesting topics. My question is to Lina as well. Lina, from clinical point of view, you know, diclofenac is used on need only, not chronically. You cannot use non-steroidals chronically because only in OA, and usually they shift to the other types of non-steroidals, which does not affect the stomach and so on, other side effects. So what is the rationalize of using this alpha-lipoic acid? When you are not using non-steroidal for acute conditions, you know, I mean, as a preventive from the clinical point of view, not from the pharmacological point of view. Yeah, I mean, when you're giving diclofenac to the patients, do you need to give them lipoic acid as an anti-inflammatory antioxidant effect, as a prophylactic from the damaging of the kidney? And at what dose? Do you have an idea about this topic? Shall I repeat the question? اسمعيني لينا؟ صوت راح عندك. هذا اسمع الأنسة.
Carmina, I think you're.
Yeah.
Did you get the point?
Sorry, Doctor,
when I'm giving diclofenac, when I'm giving Voltaren to the patients, I'm giving him just on need, not for chronic use.
I took the diclofenac as a type, as a member of non-steroidal anti-inflammatory drug. Yes, this is we can take a point of damage that can be acute for the other non-steroidal, but the diclofenac is available, so we took the diclofenac as an example for the non-steroidal anti-inflammatory drugs for the non-selective COX. Yeah.
You mean it's preventive for other non-steroids that can be used for OA and other, just to prevent the kidney damage? Do you expect that other non-steroidals, all non-steroidals may damage the kidney because acute kidney injury?
It
All
could
of them?
be. It could be. I don't know. But I took the diclofenac here, and the other non-steroidal also can damage the kidney. Yeah. But I can't say that the specific one. But according to my study, the diclofenac, it's cause acute renal failure by.
Definitely, it's well established. Yes.
Yeah.
So you need a protective drugs.
For sure, in order for the chronic treatment with the non-steroidal, like in the case of osteoarthritis and other. But the diclofenac is an available one, and it's a part of non-steroidal anti-inflammatory drug. So I took it. Yeah.
Well, we need further clinical study for this aspect,
for sure.
just to prove. Okay, thank you very much.
Thank you, Doctor.
Thank you, Dr. Manal and Lina, for answering our questions. Let's see if we have anyone else who wishes to contribute or ask anything to the participants. As for our poll, currently I see that there is a 34% participation, and so far we have from day one Canard molecular docking and synthesis of some new 5-member rings reading along with along with Lina Bahjat. Actually, she already popped up. So Lina Bahjat with 24%. She's leading by now. I'll let it be for a few more moments, and by that time I'm wishing for an increase in the poll count so that it can be fairly adjusted. And in the meanwhile, I'll go ahead and double check for the Q&A session if there are any open questions. And what I'm noticing here is that we do have a couple of questions for Dr. Brendan. Dr. Brendan, are you available to answer them if you can be able to do that?
Give me a few seconds.
Okay. Dr. Bahar has asked, is FLT3 inhibitors are effective just in 30% of those patients with AML?
Excuse me. Yeah, Harris.
Yeah, Doctor.
Okay.
Yeah, Dr. Zainab.
I'm here.
Hi, Doctor.
Hello.
Hello. I'm sure you didn't expect this comment. Okay.
Oh.
So
no, it's fine.
So we do have a couple of more questions here by Professor Dr. Bahar. I'm not sure if you're able to see them in the answer section.
Let me see. Can they ask the question now?
Let me see. I allowed Dr. Bahar to speak, but I'll read it to you if you wish to. Is FLT3 inhibitors effective just in 30% of those patients with AML? That's the first question.
So yes, typically you would use a companion diagnostic, and you would identify the patient that has the mutation that the drug can target. Right. So I think that's key in precision medicine. You have to find the patients that will respond to the medication. So not every patient with AML is a good candidate to be put on the FLT3 inhibitor. It's really going to depend on the their unique pathology of how the tumor is growing. And that's across the board for any type of precision medicine. The key is to identify the patients that will respond so that they can be given the appropriate therapy to help treat their specific cancer.
Okay, thank you. The other question is KV1 drug evaluated and proven its activity in vivo, that is in mice xenographed to assess tumor volume, or just in vitro or CADD or molecular docking?
Yeah, so the
the drug was evaluated in an in vivo xenograph model. But I think the caveat to this is that the cell line that was used, which is MV411, right, it's a suspension cell line, and we grow it subcutaneously under the skin. And we just did this primarily just to kind of show the idea that this was able to be something that could block the growth of a FLT3 driven tumor in vivo. But obviously that's a pretty poor model when you're thinking about accurately modeling the disease, right? There are better models that actually use cells that are tagged, and you inject them into the bone marrow. And then that'll be a suspension, and that's the better way to actually test. It's more clinically relevant, but we just used this subcutaneous xenograft just to show that we were able to block FLT3 driven growth in an in vivo system.
Okay, fair enough. The last question would be, what are the advantages of GCMS over other instruments like HPLC or NMR in omics studies?
For me?
I'm not sure. I'm not quite sure. I wish Dr. Bar
Oh, yeah,
would. I don't think that's related to you.
This is a question to Dr.
I don't
Lina.
know.
That's probably to Dr. Lina.
Yeah,
okay.
Yeah, sorry. Thank you so much, Doctor.
Oh yeah, thank you. And obviously, I think you all have my email. I'll put it in the chat. If there
That
are
would
any
be
other
wonderful.
questions, feel free to reach out to me, and I'll be happy to answer those questions.
Thank you so much. Okay. So I'll be going back to the polls, and let's see what we have so far. And by now, we do have a 54% of participation. So that's a good percentage, I believe. That's an acceptable one. Won't you agree? And by that, the choice of the best researchers so far have been equalized in between Rina Bahjat Qasim, evaluation of renoprotective effect of lipoic acid, and Haidar Rahim Al-Timimi, molecular docking synthesis characterization. Both have been equally got 23%. Okay, so I will be saving this. I'm not sure if you are able to see the percentages of the poll, are you?
No, we are not.
No,
We're
you're not.
unable to see.
Okay, okay. So you have to trust me on this one. Okay.
We are.
Okay. So thank you so much for all of you participants and audience for your time. It's been a long one, and we look forward to seeing you again in the next conferences that would ahead. I'll be leaving final remarks to Dr. Enham. If she has anything to add, please, Dr. Enham, if you can hear us.
Yes, 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 please. Thank you, Dr. Ara. Thank you.
Thank you.
Dear guests and all attendees, listening, discussing big topics in pharmaceutical research. In brief, I can say that this conference has been stimulative and useful in both pointing out the most important points in pharmaceutical research and in outlining important future topics in this field. I would like to say I highly appreciate all the efforts from all the speakers, our distinguished speakers. Big thank you to all the organizing committee. Big thank you to all the attendees for spending 2 days with us. And hopefully we can see you all soon in our next conference, which will be held next May, hopefully. Thank you very much and see you soon.
Thank you, Dr. Now.
أستاذ آراء.
And Dr. Munder, if you'd like to add anything, please go ahead.
السلام عليكم ورحمة الله.
عليكم السلام.
طبعا نشكر كل القائمين على هذا المؤتمر. فعلا كان الكلمات الفيدباك اللي اخذناها من زملائنا واساتذتنا في الخارج والمشاركين كان محل اعجابهم. فالشكر جزيل للجنه التحضيريه رغم انه ضيق الوقت كان برئاسه الدكتور انعام، برئاسه المبدعين الفنيين دكتور آراء واستاذ محمود، بقيه الاعضاء اللي شاركوا باللجان التقديم، طلابنا الدراسات العليا شاركوا بالبحوث، العلماء المبدعين في محاضراتهم بالامس واليوم، كانت فعلا محاضرات نوعيه. كل الشكر والتقدير لهم. احنا مثل ما قال الدكتور انعام ان شاء الله في منتصف الخامس عندنا مؤتمر حضوري. يمكن مرات نأسف نقول حضوري لان قد يكون صعب على ناس مبدعين متميزين متألقين مثل الأساتذة اللي يشاركونا بالمؤتمر يحضرون بمؤتمرنا الحضوري، بس احنا نتطلب أيضا حضوري على مود إن شاء الله الأوضاع الصحية زينة، ونأمل ونحاول نقنع بعض العلماء المبدعين الباحثين من شتى الدول الغربية والعربية المشاركة ويانا. أكرر شكري لكل القائمين والمساندين والمستمعين والمشاركين في هذا المؤتمر، والسلام عليكم ورحمة الله وبركاته.
وعليكم السلام.
وعليكم السلام ورحمة الله وبركاته.
وعليكم السلام ورحمة الله.
Thank you, Dr. Munder. Thank you, Dr. An'am, Ara, and all participants and the organizing committee for inviting us to this scientific international meeting. It was very well organized, very well prepared for. Thank you so much.
Thank you, Doctor.
You're welcome. More than welcome, Dr. Zainab, Dr. Brandon, all the attendees. Thank you so much.
Thank you and have a good day, everyone. See you around soon.
Thank you.
Bye-bye.
Bye.